Good day, everybody. Magandang umaga sa ating lahat. I am Shilette Monzon of the Philippine Genome Center's Communications team. I will serve as your moderator for today. We would like to welcome everybody to our webinar entitled Focusing on Philippine Biodiversity, the Philippine BioGenome Project. This is brought to you by the Philippine Genome Center's program on biodiversity, ethnicity, and forensics, and also by the UP Diliman's Institute of Biology. Thank you for joining us today. What do we have in store for you, you might ask? We have a list of our local scientists who will be presenting their ongoing biodiversity research using techniques in whole genome sequencing. Aren't you excited? For our friends and colleagues online who are not able to register, we are also available through live viewing in Facebook. Please visit PGC's Facebook page. It's PH Genome. You may also find the live stream linked in our Zoom chat. You can share it with your friends. Today, to officially start the program, let me call on our program director for biodiversity, ethnicity, and forensics, Dr. Maria Corazon de Ungria. Take it away, ma'am.
Hello, good morning. Can you hear me?
Yes, ma'am.
Oh, yes, okay, great. Thank you. All right, so good morning to everyone. Of course, to our distinguished guest, VP Cynthia Bautista, and all our other speakers. Magandang umaga po sa ating lahat. So now, this would be the first webinar of the program on biodiversity, ethnicity, and forensics for 2022. Of course, I see Dr. Ian Fontanellia, our always masigabong partner from the UP Diliman Institute of Biology. So first of all, I'd like to welcome everyone. As I've said, the Philippine Genome Center, being focused on genomics, is undertaking another worthwhile, very important program, which we want to discuss with you this morning. The program on biodiversity, ethnicity, and forensic explores the unique biodiversity in all living organisms, or as you will hear later, or once alive. Meron tayong speaker on archaeogenomics, and you'll hear that a little bit more later on. For our opening remarks, syempre, we want to hear the voice of our very own Vice President sa UPC, University of the Philippines, and our distinguished guest. And I'll introduce her to you now. VP Cynthia Bautista is a professor in sociology, a former dean of the UP Diliman's College of Social Sciences and Philosophy, a former executive director of the UP Center for Integrative and Development Studies, and commissioner of the Commission on Higher Education. VP Bautista obtained her BA degree in sociology from UP Diliman, magna cum laude, and later earned her MS and PhD degrees in sociology at the University of Wisconsin in Madison. Her awards are many, but I'd like to point out two particular ones, which are 10 Outstanding Women in the Nation's Service, and the National Research Council of the Philippines Achievement Award for the Social Sciences. Ma'am VP Cynthia, when I was trying to figure out how to introduce you, bigla na lang po ako napatula. So I'll read you what I wrote about 20 minutes ago, and so we can start with today's webinar. Katuwaan lang para we start light. Hindi man biologist si VP Cynthia, pero siya ang magpapasimula ng usaping biodiversity na may ugnayan sa Philippine ethnicity. Programang napakaganda ang pag-uusapan ngayong umaga. Pagmamahal sa ating kalikasan ay ating taimtim na pakinggan. Good morning, everyone, and take it away, VP Cynthia.
Thank you very much, Cora. Ang ganda naman. Picard Executive Director Ray Ebora, PGC Executive Director Cynthia Saloma, Institute of Biology Director Ian Fontanellia, and of course, Director of the Philippine Genome Center's Biodiversity, Forensics, and Ethnicity Program, Cora de Ungria. The speakers in this webinar, colleagues in the University of the Philippines system, in various government agencies, and participants with interest in biodiversity conservation research, related research, and policy. A pleasant morning to all of you. It is my honor and pleasure to formally open, on behalf of the University of the Philippines, this webinar on the establishment of the BioGenome project hosted by the Philippine Genome Center and the UP Institute of Biology. Almost 30 years ago, the University Center for Integrative and Development Studies, the UP Systems Policy Research Unit, deviated from its usual policy-oriented research to establish the Biodiversity Conservation Program. The premise of this deviation is it would be difficult to formulate appropriate policy recommendations without a taxonomy of the country's flora and fauna, with special emphasis on those endemic to the Philippines. Co-convened by the late Edgardo Gomez and Priscilino Zamora, and convened later by the late Perry Ong, the mandate of the Biodiversity Conservation Program then was precisely to complete the identification, description, and classification, particularly of endemic flora and fauna and microbial species, and ensure that they are properly conserved and protected from, among other threats, being smuggled out of the country. Having worked closely with the conveners and fellows of this program in organizing various forums and conferences as the center's executive director at the time, the webinar I'm welcoming you to, which underscores the idea and value of whole genome sequencing of taxonomic units, was unimaginable then. Since that time, the very rapid pace of development in DNA research and omics technology has opened up possibilities for the Philippines to maximize the benefits of discoveries in the omics sciences in search of creative solutions to societal problems, including serious threats to the country's biodiversity. It has also made it imperative for us to link with global projects that share the same philosophy, vision, mission, and goals. Against this backdrop, the Biodiversity, Ethnicity, and Forensics program of the Philippine Genome Center, which, as you all know now, is a genomics-focused multidisciplinary research unit of the UP system, explores the unique biodiversity in all living organisms, as Cora mentioned earlier. This program initiated the Philippine BioGenome project that aims to establish a national collaborative network of universities and colleges to sequence and catalog the complete genome of selected Philippine taxa, prioritizing, as the late academicians Pri Zamora and Ed Gomez and Perry Ong did, Philippine endemic and threatened species. What is remarkable, however, is that the project will data mine genome sequences to discover new information about Philippine species towards formulating science-driven policies on biodiversity and conservation, agriculture and food security, forensics and environmental protection, health and natural therapeutics, and ethnicity and human origins. This project is also our country's participation in the Earth Biogenomes Global Initiative. Today's webinar will concentrate on the biodiversity of some Philippine flora and fauna, as shared by our researchers in Luzon, Visayas, and Mindanao, which will certainly be enhanced, with proper support, of course, by the sequencing of the entire genome of carefully selected taxonomic units, such as the Philippine spotted deer, that is found only in Western Visayas and which has been successfully bred in captivity, crocodiles, bats, and the Philippine Eagle, which have been included in the various research initiatives in PGC Mindanao, mollusks, an organic source of biochemicals that could be used in the pharmaceutical and cosmetic industries in UP Baguio. The webinar will also share the interesting work of the Institute of Biology in developing an animal forensics laboratory that can provide very powerful genetic tools to police the trafficking of our wildlife, as well as the exciting study of chickens, not only for breeding and meat production, but also to help understand Philippine culture and human migration in the Asia-Pacific region. As genomic research advances, and it is advancing very fast, and our university is very supportive because this is one of the frontiers that we should be in. We hope that our graduates are enabled to participate and engage in projects such as the Philippine Genome, a biogenome project. For them to be able to do so, however, we in the university may need to hone competencies that would require a purposeful re-examination of the curriculum and our modalities of teaching and learning. Apart from appreciating the value of whole genome sequencing and biodiversity conservation, may this webinar also stimulate thinking about the content and modes of delivery of the curriculums of disciplines and interdisciplinary fields that have contributed perspectives, methodologies, and policy formulations towards biodiversity conservation. May you all have an enlightening webinar, and good morning and a warm welcome to all of you.
Marami pong salamat, Ma'am Cinch. Before we let you go, let me present a certificate of our appreciation for always supporting PGC in every project that we do. You are one of our staunchest allies for the whole genome sequencing technology. Marami pong salamat. At gusto ko rin pong banggitin na si Doc Cora, bago po nung inintroduce po niya, gusto ko lang po malaman ng lahat na si Doc Cora ay part of our organizing committee para sa webinar na to. At sobrang excited po namin na ibahagi itong webinar para sa buong Pilipinas. And I'm mentioning that because right now we have 400 participants, all from Luzon, Visayas, and Mindanao. So I'm so excited. To continue, marami pong salamat.
Thank you.
Ayan. To continue, nabanggit po kanina na we will introduce one of our bigger projects in PGC, the Philippine BioGenome Project. This is a research initiative, ulitin po natin, spearheaded by PGC's Program on Biodiversity, Ethnicity, and Forensics. And it also aims to establish a national collaborative network of universities, research institutes, and agencies. So magandang sign po yan na madami pong nag-attend ng webinar today. So before we open the webinar, we have another keynote speaker. Gusto po namin tawagin si Dr. Reynaldo V. Iborra, the current Executive Director of DOST's Philippine Council for Agriculture, Aquatic, and Natural Resource Research and Development of the Department of Science and Technology. As the top executive, Dr. Iborra leads the Philippine National Agriculture and Resources Research System in the formulation of framework, thrusts, and programs for the development of the agriculture, aquatic, and natural resource sectors, as well as the generation and allocation of funds for this purpose. Dr. Iborra obtained his PS Agriculture degree, major in entomology, and master's degree in entomology from UP Los Baños. He completed his PhD in entomology at the Michigan State University, East Lansing, Michigan, USA. Let us all welcome Dr. Iborra.
To Dr. Cynthia Rose Bautista, UP Vice President for Academic Affairs, Dr. Cynthia Saloma, Executive Director of the Philippine Genome Center, Dr. Maria Corazon de Hongria, Director of the Program on Biodiversity, Ethnicity, and Forensic under PGC, to our partner agencies present, and to our researchers and students joining us today. Warmest greetings and good morning. Magandang umaga po sa ating lahat. On behalf of the Philippine Council for Agriculture, Aquatic, and Natural Resources Research and Development of the Department of Science and Technology, or DOST-PCARD, I would like to congratulate Dr. Maria Corazon de Hongria and her staff for their efforts in pursuing this biodiversity webinar to showcase the initiatives of the Philippine BioGenome Project. The Philippines' terrestrial and aquatic ecosystem is considered a paradise of biodiversity, serving as a habitat for a myriad of rare wildlife, some found nowhere else in the world. With this, PCARD developed the Biodiversity S&T program with the overall goal of biodiversity conservation and management, sustainable use of biodiversity resources through product development, considering the fair and equitable sharing of benefits, and improve production and breeding protocols of important biodiversity resources. PICARD's Biodiversity S&T program focus on researches related to resource assessment, development of new and existing technologies and protocols on biodiversity conservation and management, and valuation of biodiversity resources both in terrestrial and aquatic ecosystems. R&D projects supported by the program have led to the discovery of new plant and animal species, as well as habitat and new distribution records of threatened species, establishment of protocols on the economic valuation of biodiversity resources and ecosystem services, development of new biodiversity-based products and technologies, documentation of indigenous knowledge on the use of biodiversity, and establishment of research facilities for biodiversity conservation. PICARD has always been a partner of the University of the Philippines, Diliman, in order to generate R&D plans on projects in pushing the best of the technologies in biodiversity conservation and sustainable utilization. Some of the technologies developed from R&D initiatives are on conservation using genomic-assisted tools, biological resource, bioprospecting and benefit sharing, genetic engineering, nanotechnology, and data management. We've also had projects on genomic characterization and DNA barcoding for sustainable biodiversity conservation. To date, PICARD is continuously supporting and funding various research and development projects on terrestrial biodiversity, inland aquatic biodiversity, and marine biodiversity. These projects have provided science-based, appropriate, and realistic solutions to the identified problems on the country's biodiversity. The crops and forestry sectors under PICARD's Biodiversity S&T program focus on promoting biodiversity conservation and development of biodiversity-based products. This includes conservation of the agricultural genetic resources of Philippine native plants, protection of Philippine flora and fauna, application of ex situ and in situ conservation strategies, and assessment of potential ecotourism sites. R&D projects supported by the program have led to the discovery of new plant and animal species as well as habitats, and new distribution records of threatened species, establishment of protocols and the economic valuation of biodiversity resources and ecosystem services, development of new biodiversity-based products and technologies, documentation of indigenous knowledge on the use of biodiversity, and establishment of research facilities for biodiversity conservation. This includes inventory of tamarao and other wildlife in Mount Calabiti Wildlife Sanctuary and the development of genetic groups of Philippine native pigs and native chickens through an organized breeding and selection system to ensure conservation. For the inland aquatic resources under PICARD's Biodiversity S&T program, the focus is on the sustainable management of the ecological services of the Philippine lakes. Particularly, the program focuses on understanding the contribution of Philippine lakes to the global natural emissions of greenhouse gases, developing marginal aquatic resources as sustainable habitat, and restoring indigenous and valued endemic resources for biodiversity enhancement. This also includes the development of propagation protocols for conservation of freshwater resources such as ludong and the threatened catfish. Marine biodiversity under PICARD's Biodiversity S&T program aims for the conservation and management of the genetic, species, and ecosystem diversity of different life forms found in the marine environment, such as marine plants, animals, and microorganisms, as well as the genes that they contain and the ecosystem that they form. In order to realize the ultimate goal of marine biodiversity conservation, which is the sustainable utilization of marine resources, activities such as resource and ecosystem assessment and monitoring are being conducted. New species of both terrestrial and aquatic flora and fauna have been discovered through R&D. Terrestrial species of flora includes 3 new species of native plants in 3 key biodiversity areas of Cebu. 2 species are ground orchids and 1 species is a vine. Aquatic species include new species of reed algae and new species of freshwater sponge. A refugia for rare and endangered species of corals and fishes in the deep seas was discovered through the project implemented by UP Diliman. There were also new distribution records of native trees and wildlife fauna in the key biodiversity areas of Cebu. In Mindanao, critically endangered plants endemic to the Philippines have been recorded. New biodiversity-based products and technologies have also been developed, some of which were local natural fungicide from a shrub called Tasmania piperita, botanical pesticides from indigenous plants of Central Luzon, 6 new genetic groups of Philippine native pigs, breeding of signature goat population in Region 2, 2 pure lines and 1 commercial hybrid line of Itik Pinas, and genetic breeds of Philippine native chickens. Research facilities for biodiversity conservation were likewise established or upgraded. Particularly, the first ever spore bank in the country was established in Bukidnon. Also, the first ever forest canopy observation network in the Philippines, used for the scientific exploration of forest canopy, was established in Mount Makiling, Los Baños. A molecular laboratory in CTU, devoted for the study of genetic diversity of endemic flora in Cebu Island key biodiversity areas, was equipped for DNA barcoding experiments. Living collection of diverse threatened Philippine plants was established in Makiling Botanic Garden's greenhouse and laboratory. Makiling Botanic Garden, virtual garden, and program websites were also developed. With our collaboration in the past and this present undertaking, DOST Picard will continuously commit in heeding the call of improving people's lives through science and technology. We always look forward to support our partners in developing more programs and projects in order to promote and raise consciousness on the role of science and technology in our daily lives. This undertaking is specifically an opportunity to share the importance of whole genome sequencing of Philippine biodiversity towards conservation, trade surveillance, health, and food security. Hence, I am pleased to underline our support to your institution in all your endeavors. Again, thank you very much for inviting us in this event, and may the exchange of knowledge in this activity deliver lasting impacts. Magandang umaga pong muli sa inyong lahat at maraming salamat.
Thank you for that wonderful message, Dr. Ebora. Let me now present our certificate of appreciation for your very kind, sincere words. And I'm pretty sure our audience right now is excited to hear our invited resource speakers. But before we welcome our first guest expert, please note that you may submit questions for all of our speakers in the Q&A tab during our speakers' presentation. The Q&A tab is available for all our Zoom participants. And we will be holding an open forum later to give the speakers a chance to address any questions you all might have from their talks. Now, allow me to introduce our first speaker. Dr. Ian Kendrick Fontanellia, or Dr. Fontanellia, is the director of UP Diliman's Institute of Biology and has earned the faculty rank of Professor 8 and the title of UP Scientist 1. He received his PhD in genetics at the University of Nottingham in UK in 2010. His specialization includes molecular phylogenetics, evolution, wildlife forensics, and malacology. He is currently the head of DNA barcoding laboratory in the Institute of Biology, where he and his team generate DNA barcodes for Philippine endemic species to populate the database of Philippine flora and fauna. Let us all welcome Dr. Fontanellia with his talk entitled Establishing the Earth BioGenome Project, the Philippine Network.
Thank you very much, Shalette. Thank you everyone for attending this very important webinar. It is our hope that after this webinar, we will be able to forge partnership throughout the Philippines for this very, very important endeavor. So I hope you can see the presentation now. So our goal for today is to give an idea or give focus or emphasis on the importance of establishing a consortium that focuses on obtaining the whole genomes of our endemic species. So, of course, for those of you, I might be preaching to the choir here for some of you, but of course, for many of you, we know that the Philippines has a very high biodiversity, especially in terms of endemicity. When we say endemicity, the presence of specific taxa only in the Philippines and nowhere else. Now, just to give some perspective with regards to birds, for instance, we have about 683 known species found in the Philippines, 35% of which are found only here in the Philippines. In terms of amphibians, we have the highest, we have a very high endemicity of about 84%. So if you're going to look at the terrestrial vertebrates found here in the Philippines, and when I say terrestrial vertebrates, I exclude humans or Homo sapiens. We have about 47% endemicity. That's nearly 1 in 2 species found in the Philippines that are not found anywhere else. And of course, we also have what you call, we also have what you call the super endemics, which are found only in certain areas of the Philippines and nowhere else. Some of them may be due to historical distribution of these species. Others may be due to habitat encroachment because of human activity. Now, some examples of these super endemics include the tawilis, Sardinera tawilis, found only in Lake Taal. And then we also have the Isarog shurat, Rhinchornis isarogensis, found only in Mount Isarog. And then, of course, we have the Cebu black shama, which is found only in Cebu. Others include the Bubalus mindorensis, found in Mindoro, and then you have the Manus polionensis, also found in Palawan, and then the Kakatuwa or the Katala, which used to be found in many islands of the Philippines, but are now unfortunately restricted to the island of Palawan. And then we have also the Rusa alfredi, or the Visayan spotted deer, found in Western Visayas, and our iconic national bird, the Philippine Eagle, which are found in several islands, including Mindanao, Samar, Leyte, and some parts of Luzon. But many of these species, in fact, all these species are endangered, and there is a need to protect them. And it is unfortunate that in terms of endemicity, these are our country's national resources, and yet we do not have yet their genomic resources. We don't have their whole genome sequences. Okay. Now, the Philippines has a very high biodiversity. So in terms of plants, we have around 3,800 to 6,000 endemic species. For marine species, corals, for instance, we have 421 out of 577 known species in the world. They are found here. So here are some examples of vertebrates, some vascular plants, and of course, my favorite group, the terrestrial snails, because I'm a malacologist. Now, on top of having a high biodiversity, the Philippines is a biodiversity hotspot. Now, what is a biodiversity hotspot? This is a relatively small area with a great concentration of endemic species, and many of these endemic species are threatened because of human activity. So as you can see here, the entire Philippines is a red, is a pulang-pula, magliliyab sa kapulahan, because it is a biodiversity hotspot. In fact, it is a biodiversity hotspot amongst biodiversity hotspots in the world because of the high rate of extinction. Kasi nga, many of the habitats of our endemic species are rapidly being encroached by humans as a consequence of our rapid population growth. Now, there are 3 levels of biodiversity in which we can measure the richness of our biodiversity. Of course, we can look at it at the molecular level. So this is what we call the genetic diversity. And then we also have species diversity, which is simply counting how many species are found in a particular area or across the Philippines. And then we also have what you call ecosystem diversity, which also leads to high biodiversity in the Philippines because we have a rich biodiversity in terms of different ecosystems. So meron tayong mga ecosystem niche where different species can thrive, therefore supporting a myriad of different species. each with a specific ecological condition, set of ecological conditions wherein these organisms can survive. Now, if we focus on genetic diversity, the ultimate measure of variation and genetic diversity can be measured at the level of the DNA and the genome. Now, you may ask, why? Why do we need to sequence the whole genome of a particular organism? Well, from the scientist's perspective, why not? But for the general population, for the general public, there are important reasons why we should sequence life. One, it will revolutionize our understanding of biology and evolution. That's one. So that's basic biology. Secondly, and this is particularly true for many of our endemic species, the information that we will get from the whole genome sequence can help us conserve, protect, and even restore biodiversity. And third, and if we're going to be anthropocentric about it, we can create new benefits for society and human welfare. So, malaki rin ang tulong na maidudulot dito sa atin mismo if we have these biological information, genetic information available. Now, here are some specific examples as to why we should sequence whole genomes. For humans, syempre, isa sa mga unang na-sequence na whole genomes ay yung human. And that's why we had in the late 90s the Human Genome Project, which cost billions of dollars just to get one set of one haploid set of the human genome. That was more than 20 years ago, but nowadays it would cost even less to whole genome sequence one individual. So for humans, we can use the information for disease diagnostics, disease risk, human ancestry, which Dr. De Ungria is working on, and also forensics pala. And then livestock and wildlife for disease diagnostics, genome selection for milkweed, for example, and parentage control, conservation of endangered breeds and species, etc. For plants, you have genomic selection to improve crop yields and other agronomically important traits. For biofuels production, gene editing, conservation of endangered species. For insects, we can use them for gene drives, genome editing, and pest control. For fungi, we can use them for synthetic biology, metabolic engineering for drug production and useful chemicals, biofuels production, etc. And bacteria, so microbiome in health and disease by processing, detection, surveillance, and host response, genome epidemiology, and understanding microbial diversity. And viruses. We all know that viruses are technically not living organisms, but they do carry genomic material, either in the form of DNA or RNA. And we have just realized in the past 2 years the significance of knowing the whole genome sequence of viruses, in particular COVID-19. So SARS-CoV-2 rather. And because of our ability to determine the sequence of SARS-CoV-2, we know the different variants available. We know how to, and we also are able to use them for monitoring and surveillance, among others. Okay? So what is the current state of whole genome sequencing in the Philippines? We know that we have a very rich biodiversity, pero ilan na ba ang ating na whole genome sequence? Well, at the moment, we only know of 3 species available in literature. So you have the tarsier, Tarsius siricta, which was a project spearheaded by a group of foreigners way back in 2016, Schmitz et al. In 2016. And then, of course, we have the Cocos nucifera, particularly the Katigan dwarf of coconut by Lantican et al. In 2019, and Musa textilis or abaca of Galvez et al. In 2021. These are our colleagues from UP Los Baños. So, ito lang, as far as we know, ang 3 species collected in the Philippines, found in the Philippines, that were whole genome sequenced. We do, however, have a lot of animals that have been whose whole mitochondrial genomes have been sequenced. So, yung mitochondrial genome pa lang, but not the nuclear genome. So, you have several here, ranging from Tarsius, Risota to Rosetus. Okay. Now, this is where the Earth BioGenome Project comes in. Now, what is the Earth BioGenome Project, or EBP? Now, this is a moonshot for biology that aims to sequence, catalog, and characterize the genomes of all of the Earth's eukaryotic biodiversity over a period of 10 years. So this started in 2018, and the 10-year period is supposed to end in 2028. So gusto nilang i-cover at least yung eukaryotic multicellular biodiversity. So hindi na muna kasama dito yung mga protozoans, hindi muna kasama dito yung mga monerans or bacteria. So 10 years ang kanilang target. But if you will notice, in the Philippines, we only have 3 known species na whole genome sequence. At puro mga hayop pa yun. Wala pa doon yung mga fungi, wala pa doon yung mga halaman. So we have a long, long way to go. And in fact, we are not yet actively a part of this endeavor. Now, this EBT aims to produce an open DNA database of genomic sequences that provides a platform for scientific research and biodiversity conservation efforts. Now, this is led by 3 people. We have Harris A. Lewin of UC Davis, who is the chair of this project. And then we have Dr. Jean Robinson of the Carl Wood Institute for Genomic Biology at the University of Illinois, and W. John Kress, a research botanist and creator of the Smithsonian Institution. Okay. Oops. So what are the goals and anticipated outcomes of the Earth BioGenome Project? So there are 3 main goals. One is to revise and reintegrate our understanding of biology, ecosystems, and evolution. So basic knowledge of our understanding of biology, if we already is what we can get or glean if we have the whole genome sequence of many taxa. So it will provide better understanding of evolutionary relationships among all known organisms, fully elucidate the timing, origin, distribution, and density of species on Earth, and then generate new knowledge, ecosystem composition and functions, discover new species, because the ultimate method of delineating species, one of the best ways to delineate species is to look at the whole genomes and not just specific sequences. It's better to look at the whole genome and then discover fundamental laws that describe and drive evolutionary processes. Now, the second goal is to enable the conservation, protection, and regeneration of biodiversity. So we want to determine the role of climate change on biodiversity. So remember, no man is an island, and no species is can ever stand outside its environment. We always interact with our environment. Next, fully elucidate the timing, origin, and distribution. Clarify how human species, activities like pollution, habitat encroachment, etc., and invasive species affect biodiversity. When we say invasive species, these are species that are accidentally or intentionally introduced by man into an area where they are not originally native or originally found. So we also want to develop evidence-based conservation plans for rare and endangered species. And the next set of speakers will focus on this, and then create genomic resources to restore damaged or depleted ecosystems. And then number 3, to maximize returns to society and human welfare. So ito yung tinatawag natin ecosystem services and biological assets. So ano naman ang nakukuha nating ecological or economic benefit if we obtain the whole genome sequences of these taxa? Well, we can use them to discover new medicinal resources for human health, enhance control of pandemics, identify new genetic variation for improving agriculture, discover novel biomaterials, new energy sources and biochemicals, and improve environmental quality. So I will not discuss these in detail, as my colleagues will discuss these goals and anticipated outcomes using their specific taxa. Now, currently, the Earth BioGenome Project has 48 affiliated project networks around the globe. So there are 48 ongoing projects focusing on global taxa. For example, in Chile, they have the 1,000 Chilean genomes. So they have identified 1,000 taxa wherein they will get or obtain the whole genome. And this is in Chile. And then you have the 1,000 fungal genomes, the 5,000 insect genomes project. And then you have the 10,000 bird genomes, the 10,000 plant genomes, and the Africa Biogenome project. So tayo, the Philippines, we are not yet an active member we are not yet actively participating in this endeavor, despite the fact that we have a very high biodiversity. Okay. Now, EBP has 45 institution members around the globe. Now, as one of the 17 mega biodiversity hotspots in the world, the Philippines should be at the forefront of whole genome sequencing of endemic species. Because remember, as a biodiversity hotspot, we are losing many of these species, some of which we have not yet fully described. And so it is so sad that even before we can learn or understand these new species, we are going to lose them. And it would be such a shame to lose such rich information from the organismal level to the molecular level, and how these species can benefit us ecologically as well as economically. So there is a strong impetus for us to whole genome sequence these taxa even before they can disappear, in the hope that we can A, protect them, conserve them, and B, well, benefit from them. So this is the EBP organizational model. They have an EBP coordinating council, which is involved in standards, analysis, informatics, communication, sampling, and sourcing. And then you have here in the red circles, these are the different whole genome sequencing projects, which could be national in scope or global. And these are the major players. Okay, so you have the European Union, Brazil, USA, Africa, Canada, Japan, China, and Australia. So the ASEAN and the Philippines in particular, we are not yet an active member of this endeavor, which I'm sorry if I keep on reiterating this, but it is important for you to understand that we are at the center of the center of biodiversity in our region, and yet hindi tayo actively nagpa-participate. Oops. So these are the general workflow for genomics biodiversity studies. So I'll just give you a brief overview. Of course, in any research endeavor, you must have a a draft research proposal that you need to submit. And then secondly, since we have Republic Act 9147, we must coordinate with LGU and national agencies, including the Department of Environment and Natural Resources, particularly the Biodiversity Management Bureau, and the Bureau of Fisheries and Aquatic Resources to secure gratuitous permit, prior informed consent certificate or clearance, transport permits, and more important, ito madali natin nakakalimutan, but we must reiterate this as well, the National Council for Indigenous Peoples, especially if the endemic species that we are working on are also found amongst indigenous peoples and their protected areas or ancestral sites. And then for human resource development, we must also hone the skill set of our researchers or personnel that will be working on this endeavor by promoting hands-on training courses and coming up with degree programs that will focus on whole genome sequencing and bioinformatics. And then on top of that, we should also have a biobank and biorepository infrastructure where the specimens will be submitted as repositories. For example, the UPLB Museum of Natural History, the Philippine Genome Center in UP System for the DNA samples, and even the National Museum. And then nucleic acid techniques. Once you have the samples, of course, we have to extract the DNA, purify, quantify, and amplify specific regions. And this will require specific equipment. And then, of course, we proceed to DNA sequencing, which includes PCR purification, quantification, library preparation, and whole genome sequencing. We do have the technology already here in the Philippines to do whole genome sequencing, particularly to the Philippine Genome Center. And we have realized the significance of such facilities as of being able to sequence whole genome sequences, especially during the pandemic. So hindi na dapat natin kine-question pa ang rationale of having such facilities here in the Philippines. In fact, such facilities are now essential. Okay, so we also have bioinformatics analysis, which includes data processing and analysis of genome, and such as genome annotation, phylogenetic analysis, genome assembly, etc. And of course, publications and returning to local communities. So we report to publication, data sharing, and it's also important that we go back to the local communities where these specimens or taxa are found in order for them to feel their sense of ownership to these taxa. Explain to them the significance of obtaining sequences, and sila din dapat sana ay maka-benefit sa ating undertaking. So remember wag lang natin isip yung national, pati yung local scope. Sana yung pinagkukuhanan natin ng mga taxa ay makikinabang din sa ating ginagawa. So, eto lang just to give you some idea how much it costs. So, for example, the Philippine Eagle, which is around 1.28 GBP, for a maximum number of 6 sample runs, it will cost you 432,000 pesos na lang, compared to more than several $billion in more than 20 years ago just to get one human sample genome. Yung Philippine Oak Tree, which is 1.17 GBP, around 436,000 din. Yung Tarsius Siricta, slightly a little less. You have 397,000. This is based on the Philippine Genome Center DNA Sequencing Core Facility, but as a caveat, prices are subject to change dahil nga meron tayo mga, nagtataas din yung presyo ngayon, so may inflation din nagaganap. So for capacity building, we do have the Philippine Genome Center, the main headquarters located here in Diliman. So I have to make a strong pitch for PGC here. And then we also have satellite offices and facilities across the Philippines. We have one in UP Los Baños, which focuses on agriculture. We also have UP Mindanao and UP Visayas. And they are slowly but surely building up on facilities so that some of the basic molecular work like DNA extraction, tissue amplification, etc. can be done in these facilities. So hindi na po tayo centralized na lang dito sa Quezon City. Some of the work can also be done in these facilities. So another way to capacitate the Philippines, aside from infrastructure development, is of course manpower development. Now, there is already an MS and PhD bioinformatics. There are already the MS and PhD bioinformatics programs being proposed and are already being developed. And it is hoped that they will be offered in the UP system as early as first semester of 2022-2023. So, umuusad na po yung proposal na to. And please, if you have anyone interested, for MS Bioinformatics, it is 36 units, 2 years for full-time. I must reiterate, full-time para maka-complete ng maka-complete ng degree. And in PhD Bioinformatics, it is around 37 units, 3 years full-time if you have an MS, and probably 61 units or 4 years full-time if you only have a BS. So pwede kayo mag-straight PhD, at least based on the proposal. So to summarize, what are the challenges here in the Philippines? There are limited data on Philippine endemic species. We only have 3 known species wherein we have whole genome data. There is need to effectively communicate the value of whole genome sequencing, and I'm very glad that we have a very strong participation today. As of now, we have 472 participants, and it is my hope that you will be able to communicate to your local partners, wherever you may be, the significance of whole genome sequencing of endemic species. Of course, we must practice ethics in doing research, and that means including getting the proper permits. And then there's also a need to utilize museums and biobanks for storage of biological materials. I can't emphasize enough the need to partner with our museums, particularly the National Museum, because they also have in storage endemic species, and maybe we could also tap them for whole genome sequencing. Prospect, there's a huge opportunity to work on a diverse group of taxa within the next several generations. Marami tayong mapoproduce ng mga undergraduate and graduate students na pwedeng mag-work dito sa ating whole genome sequencing undertaking. We must form collaborative networks within the Philippines and outside with the Earth Biogenome Project. And there is a strong opportunity for human resource development in the field of bioinformatics, genomics, and other omics na pwedeng manganak sa genomics. So you have transcriptomics, proteomics, glycomics, etc. So ayun, maraming salamat sa inyong lahat.
Thank you, Dr. Ian, for that wonderful introduction of the talks and for also promoting whole genome sequencing and encouraging people to actually go and study for their PhD straight na, no? But before we let you go, we would like to present this certificate of appreciation for you. Or actually, sir, I would like to commend you as part again of our organizing committee din. Dahil palagi kang nandyan. Okay? Maraming salamat, Sir Ian.
Salamat.
Anyway, let us go to our second speaker for today. Let me introduce Professor Leif Erickson Gamalo, a faculty member of UP Mindanao and staff also of our satellite PGC in Mindanao. He received his MS in zoology at UPLB in 2018. He is interested in studies concerning wildlife biology and conservation, primatology, wildlife parasitology, and human-wildlife interactions. He has a very interesting talk today, actually. I really wanted to listen to his talk. It's all about the whole genome of Philippine eagles and other taxa from Mindanao, an initiative to utilize genomics for critical conservation decisions in southern Philippines. Let us all give a virtual hand for Professor Leif Gamalo.
Hello, good morning po. So I will be presenting this topic, understanding the whole genome of Philippine eagles and other taxa from Mindanao, an initiative to utilize genomics for critical conservation decisions in southern Philippines. Okay, so let me start my presentation by introducing yung different threats sa biodiversity sa region. So of course, yung number one dyan, yung habitat destruction. Meron ding hunting na nangyayari. For example, sa long-tailed macaques and other bird species na kadalasan din ay ginagamit for both local and international trades. An example, pet trade or for biomedical research studies. The presence of invasive species din for both flora and fauna, climate change, and diseases. An example dito yung na-observe namin na mass die-off of Philippine warty pigs sa isang forest sa Tagum City. And yung dahilan nun ay yung African swine fever. To address these issues and threats sa biodiversity, of course, kailangan natin ng conservation activities and mga scientific studies para mag-support ng decisions for conservation. So actually, marami nang ginagawa din sa Mindanao for the protection of biodiversity. And sa UP Mindanao, meron din kami mga programs and projects. Example, yung Human Macaque Interface Program, kung saan gusto namin ma-understand kung ano yung interaction ng tao at yung unggoy at yung kaniyang habitat sa selected places sa Mindanao and Leyte Island. Meron din tayong NICER Vatas Project. Gusto naming malaman kung ano yung mga viruses na meron yung mga wildlife, yung domesticated animals, and yung tao at kung may nangyayari ng palitan ng viruses, particularly coronaviruses. Another one, itong Urban Biodiversity of Davao Project. So, gusto namin dito i-highlight na yung urban landscapes ay pwede pang tirahan ng biodiversity. And I think very helpful siya for urban planning and the Iho Forest Project. Dito namin pinag-aaralan kung ano ba yung effect kung mawala ang isang very important species. Particularly mga ecological engineers katulad ng baboy ramo na naubos dahil sa African swine fever. Aside from field-based projects, through the Philippine Genome Center Mindanao ay gumagamit na rin kami ng genetics and genomic studies for biodiversity studies and conservation. So example dito, itong mga publications, DNA barcoding on selected fruit bats in Davao City using fecal samples. Nagsasabi dito sa paper na pwede tayong maka-identify ng species ng bat using yung feces nila. And even, pwede rin makapagbigay ng information sa phylogeny ng species. And aside from barcoding, ay gumagawa na rin ng mga genomic studies. For example, itong first complete mitochondrial genome sequence of Cynopterus brachiotis from the Philippines. And ito yung isa sa mga results. Nagsasabi lang dito na yung Cynopterus brachiotis natin ay genetically different from Cynopterus species from other countries. At itong genomic data ay nagsosupport sa mga past genetic studies na yung ating Cynopterus brachiotis is a possible different species sa mga Cynopterus brachiotis from other countries. Okay? Maliban sa mga publications dito, currently, ang PGC Mindanao ay meron ding mga ongoing projects and initiative. At isa dito ay itong Mindanao Bat Metagenome Initiative. At ito yung mga objectives. So identify the evolutionary relationship of Philippine population to other populations outside the country, contribute to the taxonomic status, and serve as an additional supporting information for other phylogenetic, evolution, and ecological studies of bat species in the world. At yung update dito ay na-sequence na yung ibang individuals. And maliban sa bats, meron ding isang metagenome initiative na currently ginagawa din. And that's the Philippine Eagle Metagenome Initiative. Very important para malaman yung gene flow na nangyayari sa Philippine Eagles sa different areas sa Mindanao. At ito yung objectives. So identify the genetic diversity of Philippine eagles from Mindanao Island and understand the evolutionary phylogeny of Philippine eagles sa Mindanao. Okay, and here comes the Philippine BioGenome Initiative. Ito, yung objective is to sequence the whole genome, not only the mitochondrial genome, but the nuclear genome also ng wildlife and other taxa sa Philippines. So in our part, ito yung aming na-recommend. Philippine Eagle, Philippine Slow Loris, Philippine Crocodile, Mindanao Frog, and these are threatened species according to IUCN or DNR, and the Marcobra, which is highly persecuted sa region. So ang question, bakit kailangan i-sequence yung whole genome ng mga species na to? So for conservation, actually maraming reasons kung bakit kailangan. Maliban sa mga nasabi kanina na applications katulad ng taxonomic assignment, determining evolutionary lineages, meron pang ibang applications na makaka-affect sa decision for conservation ng mga species na to. Example, ito, identification of genetic diversity and adaptation for reintroduction purposes. Now, for example, alam na natin yung genetic information or genomic information ng Philippine eagles. So from that, malalaman natin yung mga adaptive traits na meron sila. And example ng adaptive trait ay disease resistance. At kung alam na natin kung sino yung individuals na may adaptive traits na to, ay pwedeng sila yung introduce sa wild. No, para sunod na generation, yung mga Philippine Eagles, ay posibleng magkaroon na ng mga adaptive traits na ito. Okay, another one is identification of inbreeding conditions. Yung inbreeding ay isang effect ng lumiliit na population ng wildlife, no? At hindi siya actually healthy for the species. And kung alam na natin yung conditions ng inbreeding, for example, sa captive setup, malalaman natin kung sino dapat yung i-pair for breeding programs. Another application ng genomic information ay for surveillance of wildlife and their habitats. No? So using eDNA signature, mako-confirm natin yung presence ng mga cryptic and elusive species katulad ng mga nocturnal animals, example itong Philippine Loloris, Philippine Tarsier, lalong-lalo na sa area na mahirap puntahan. So environmental DNA lang ang kailangan. Ibig sabihin, environmental samples lang, mag-collect lang ng soil, water, or even air sample, ay pwede na nating ma-confirm yung existence nila sa area na yon, which is very important din for reintroduction purposes. No? Before tayo magpakawala ng animal, ay dapat ma-confirm muna kung yung habitat na yon ay ginagamit nila. Next application ng whole genome sequencing sa biodiversity conservation, ito, no? Detecting hybrids in the wild and in captivity. So very important na ma-detect yung hybrids before reintroduction sa wild population. No? Kasi pwede siyang mag-cause ng pag-collapse ng population ng mga wildlife species. Another one, sa kanilang natural habitats, pwede siya maka-influence ng management strategies for conservation ng two sympatric species katulad ng saltwater crocodile and yung Philippine crocodile. And for the case ng Philippine warty pigs, kung saan nakikipag-hybridize na rin sila sa domesticated pigs, ay maka-influence din ang genomic information sa pag-manage ng exotic species or domesticated animals. So knowing this information, I think it's very important, no, to utilize genomics for conservation decisions and management strategies. However, kailangan pa ng funding, no? And thanks University of the Philippines Mindanao and DOST sa pag-fund ng mga current projects ng Philippine Genome Center Mindanao sa different initiatives sa biodiversity conservation. And of course, sa collaborations such as Philippine Eagle Foundation and the Iho Resource Corporation. And I think it's better na mag-expand pa ang collaborations, including yung mga local government units, different NGOs, and other institutions. Ang reason yan is wala ding boundary kasi yung wildlife. No? Kahit saan sila pwedeng makita. And of course, kung ma-optimize na yung method, and it's better to extend this initiative to other taxa, no? Katulad ng invertebrates, insects, marine life forms, and even microorganisms. That's all. Thank you very much.
Maraming salamat, Professor Gamalo. Let us present us our certificate of appreciation for your very informative talk. And for those who are asking how to get in touch with our speakers, if you want to collaborate with them or to know more about their research, you can always email Mr. Enrique Fortajada, ebfortajada1@up.edu.ph. He's the point of contact for the program on biodiversity, ethnicity, and forensics. He will facilitate the channels for you to get in touch with our esteemed speakers today. And also, for more information about PGC and its services, you can always visit our website, pgc.up.edu.ph, or email pgc@up.edu.ph for services regarding whole genome sequencing as well. Okay, so let's move to our third speaker. For today, we have Professor Adrian Luxon. He is currently an instructor at the Institute of Biology here in UP Diliman, and he has earned his MS in Biology, major in genetics. His research interest is on molecular phylogenetics and population genetics of animals and how these tools can be applied in species conservation and biodiversity. He will talk today about genomics in combating illegal wildlife trade. Let us all welcome Professor Luxon today.
For my presentation today, I probably would be, I am basically siguro this is the most direct way that genomics could help protect our biodiversity. Okay? So my title for my talk is Genomics in Combating Illegal Wildlife Trade. And I hope by the end of my presentation, I could urge our funders, our students, and our wildlife enforcers to kind of think about the potential of genomics in managing wildlife trade, persecuting illegal traders, and also, and of course, protecting biodiversity. So the illegal wildlife trade is a very lucrative business. It's actually just next to human trafficking, counterfeit narcotics, and it's actually for some markets it's actually more than the value of traded goods are more than the value of traded goods are more than the weight of gold and platinum. And of course, many of our wildlife enforcers face danger because a lot of them are probably a lot of them are killed whenever they enforce, whenever they whenever they face these illegal traders. Okay. So in our country, a lot of these cases of illegal wildlife trade are posted in news. So for example, very famously, our pangolin species, the Palawan pangolin, are shown in news being traded or being seized by wildlife enforcers. And some of these are exported to other countries for their consumption or for medicinal purposes. So here are the most recent and most major confiscations that happened in the country. And as you can see, a lot of these are endemic species. So we have the pangolins, as I've mentioned a while ago. We also have marine turtles. We have the hawksbill turtles. We have the blue-naped parrots, etc. So a lot of these have been confiscated from airports, from seaports. Okay? Some are even hand-carried by illegal traders. And it's not only whole animals that are being traded. We also have animal parts. So some of them have already been processed, processed to different objects such as handles of knives, for example. And really, the amount of illegally traded animals in the Philippines is staggering. So we know that a lot of these species are already vulnerable. There are very few in the wild, and the amount of seized material is seized materials tells us that or presents the danger of really extinction, presents the danger of extinction for these animals. To combat or to help combat these illegal trade, So we have wildlife DNA forensics, which are basically analytical techniques that are capable of providing DNA evidence to assist wildlife enforcers. And they are used to determine the species, the population, the relationship, or individual identity of a sample. And they are key investigative tool to combat wildlife crime. However, unlike the human counterpart in human forensics, standards and validated markers are lacking for many species. As Sir Ian pointed a while ago, only a few of the species have had their genome sequenced. And for wildlife enforcers, their method of dealing with illegal trade or dealing with these illegally traded samples is still through the traditional morphology or taxonomic methods. And of course, not all of them have taxonomic expertise, so they have to tap academics or other experts for validation for species name or other questions for forensics. So this is my hope. For hopefully the Earth BioGenome Project network in the Philippines could be used to empower our enforcers. So through whole genome sequencing, a lot of these molecular markers can be accessed. So for example, DNA barcodes, simple tandem repeats, the mitochondrial and chloroplast genome, single nucleotide polymorphisms, and others. So these markers will help answer different forensic questions for our wildlife enforcers. And as an example, so I'll just give you some examples. So for species identification, so we have DNA barcodes. So this is something that we have been doing in our lab. We have been building up a DNA barcode database for Philippine endemics and Philippine species. So these barcodes are very good reference sequences because they are also publicly available. They are deposited in public databases such as the Barcode of Life Data Systems and GenBank. So DNA barcodes, for those who don't know, these are short standard sequences that can be used to identify an unknown sequence. So we have different molecular markers or different DNA barcodes that depending on the taxa. For animals, it's CO1. For fungi, it's ITS. For plants, we have RBCL, MATK, ITS2. Now, these markers have been or can be used to identify species. So, for example, whole animals, those that are pets or traded as pets, zoo animals, exotic plants, or those that have already been processed. So we have body parts. So as you can see here, we have coats or rugs that are made from animal skins, or just some bear paws, or even those that are highly processed goods, such as those in meat markets, our bag of teas. So those have been processed by into objects such as knives, as I said a while ago. There, so DNA barcodes can be helpful in identifying these species. Okay, so as I've said a while ago, in our lab, we have been doing DNA barcoding, and we also have been collaborating with the Biodiversity Management Bureau in assisting them in their species identification of some of their cases. So what they do is they have their DNR offices in the different parts of the Philippines. They are sent to the Biodiversity Management Bureau, and BMB will be the one to send the samples to our lab. Okay? And one of our I guess most prominent cases with them is our paper on, or our case on the DNA on the species identification of pangolin remains. Okay. Now these samples came from several sources. So the first one is from a Chinese fishing boat that ran aground into Bataha. So in this case, they found sacks of pangolin. And what the wildlife enforcers, the PCSD and DNR wanted to know if these species are our Palawan pangolin. Okay. And there was also a case in Puerto Princesa where they were able to uncover loads of carcasses of pangolin. So in both of these cases, we were successful in identifying the species. So these pangolins from the Tubataha case were actually Javan pangolin, while these species in Puerto Princesa is our Palawan pangolin. Another way or another use for forensics of wildlife DNA forensics, okay, is the identification of geographic origin. It's basically the ability to assign a sample to a specific population. Now, for example, so a lot of our species are not only endemics, they are probably found in nearby geographic areas. For example, we have here Aquilaria comingiana, which is commonly known as agarwood. So it's not only found in the Philippines, but also in parts of Indonesia. And agarwood is actually a regulated species for the Philippines. It's still prohibited species. Basically, you cannot collect them in the wild. Okay? Now, for other countries, it may be you may need to in their laws, you may need to establish the species origin where the samples were taken in order to proceed with prosecution. So another example for the use of wildlife forensics in identification of geographic origin is in this paper by Wu et al. In 2005. Basically, what they did here, this is the application of mitochondrial DNA sequence analysis in the forensic identification of Chinese Sika deer species. In their case, the Sika deer species is actually it's another prohibited species. You cannot collect them in the wild. However, there are subspecies that can be collected or are used for medicinal purposes. So these species are bred or managed. So these are legal. For some of these subspecies are legal. Now, what they found out is that some of the animal parts that are sold in the markets are actually taken from the wild, okay? Specifically in one of their nature reserves. So with the use of of mitochondrial DNA sequence, they were able to pinpoint where these seized animal parts were taken. So another example here. So this is the forensic DNA analysis of Pacific salmonid samples for species and stock identification. So in this case, salmon species are fish species that spawn in freshwater and mature in saltwater. Now, some of the areas, some watersheds or water areas are restricted to fishing. Okay? Now, with salmon, they were able to have a database of salmonid species, or basically the gene pool of salmonid species in many watersheds, and they were able to find those that are sold in markets, that some of them have been taken from restricted areas. The last one, last application for forensics, for wildlife forensics, is the identification of individuals and parentage analysis. So the identification of this type of analysis is actually it's not as popular as species identification, okay? But still it is very useful, especially when dealing with stolen animals or traded or pet traded animals. Okay? So this type of analysis is actually very common in human forensics, if you know CSI and those other TV shows. So we know that there are STR markers that can be used to identify individuals or match a sample, okay, to a suspect. So one example for applying this analysis to wildlife forensics is here in the paper by Mucci. So this is wildlife DNA forensics against crime, a resolution of a case of tortoise theft. Basically, there was a breeder of tortoise that reported theft or reported theft to wildlife enforcers and saw that they were being sold in the market. Now, to establish this connection, so what they did was to compare the suspected stolen goods with the ones that he is breeding, and they were able to establish a familial relationship between the stolen goods and the one that he has in his breeding site. Okay. Now, this also works for DNA registers. So basically, registers are basically individuals that have been DNA profiled and then stored in a database. In this example, this is for a species of whale. So this species of whale is actually or its body parts, or it's actually consumed in the market, okay? But only those that have been registered can be consumed. Now, with DNA analysis, they were able to track some individuals or some samples in the market that were not in their database. So presumably these have been caught wild. These have been caught wild or caught illegally. All right. So I hope hopefully our wildlife enforcers, our students, our audience here was able to see the potential of sequencing the genome of our species. Now, what needs to be done to advance wildlife DNA forensics in the Philippines? So I have 4 tabs here. So first is to do whole genome sequencing of some selected species. Initially, characterize the extent of variations of the whole genome sequence, lay the foundation of our own DNA tools for wildlife forensics, and of course, establish our own wildlife forensics lab. Okay. So to start, at least for my project, hopefully we will be able to start sequencing the genome of these species. You have the Palawan pangolin, the hill myna, the Philippine cockatoo, the blue-naped parrot, the Philippine forest turtle, and a species of agarwood. Now, these species are species that have been traded, that have been highly traded based on data from DNR and PCSD. And after that, we need to characterize the extent of variations in the whole genome sequence. Basically, we want to get these molecular markers, data barcodes, STRs, and see if they can be used to answer our forensics questions. Example, kanina, species identification, identification of origin, and identification of individual and familial relationships. And of course, lay the foundation of our own DNA tools for wildlife forensics. So basically, we want to validate these markers, have other labs test these markers if they are repeatable. That is important in forensics. And of course, establish a dedicated wildlife forensics lab. So this has been a dream in the making, even during the time of the late Dr. Perry Ong. Okay? So this lab, a lot of wildlife forensics lab have already been established in many countries. For example, this is the forensics lab of the U.S. Fish and Wildlife. Okay? So they have a very extensive wildlife forensics lab. They have a lot of sections. So there's a DNA section, there's a morphology section, there's a chemistry section, etc. So all of these could help answer wildlife forensics questions. And in Southeast Asia, there have already been established partnership between some of the countries, Indonesia, Malaysia, Thailand, and Vietnam, okay, to have a DNA forensics, wildlife DNA forensics group that will help in, that will work together in curtailing illegal wildlife trade in Southeast Asia. So in the Philippines, we are still not part of this group, and hopefully, if we're able to establish our own lab, and then of course, with the help of the the help of our with the establishment of our molecular markers, we can also contribute to this group. Okay, so I think that's the last of my slide. So hopefully, upon establishment of a forensics lab, wildlife forensics lab, establishment of validated markers to help answer forensics questions, we can curtail or prevent or manage our own species, prevent illegal wildlife trade, and prosecute illegal wildlife traders. Thank you.
Thank you for that very detailed talk about the framework for conserving wildlife, Professor Luxon. And while you're there, let me present the certificate of appreciation, right? Our talks today are very timely, conservation of resources and wildlife. And I do hope that everybody is very invested in this, especially now that sustainability is what we want. So let us move on to our next speaker. Dr. Wilberto Monotilla is an assistant professor in UP Diliman's NIMBB, National Institute of Molecular Biology and Biotechnology, and also a principal investigator in Aquatic Biotechnology Laboratory. He received his PhD in Aquatic Biosciences at the Tokyo University of Marine Science and Technology. His research interests include applied phycology, aquatic biotechnology, conservation biology, and environmental science. Today, he will share his expertise in his talk entitled Microalgal Genomics on the Road Towards Nutraceuticals and Agroceuticals. It is my pleasure to introduce Dr. Monotilla. You can now share your screen, sir.
Can you see my screen now?
Yes po.
Yeah, okay. Good morning. Thank you, Millet, for the introduction. Well, this is the part of the project that we are proposing for the DOST, which is part of the BioGenome project. And thank you for this the previous speakers because I will not deal with those materials used for the identification, especially in the field of whole genome sequencing. So in this lecture or webinar, I will deal more on the introduction of astaxanthin from microalgal species, which is the objective of our project. So as you can see from the slide, we've got several abiotic factors which are involved. Then we've got to have to select the strains on which we can generate keto-carotenoid astaxanthin, and with the knowledge that we gain from the whole genome sequencing, we look forward that it can be a tool for the genetic engineering researches. And similarly, we have to look at how these untapped resources can be cultured on a large scale in order to generate the desired product. So, So just a brief background, green microalgae is an ubiquitous photosynthetic organisms, whether it's freshwater or marine water. And they can serve as the primary producers in our food chain, as what we have learned in our biology classes, and can be used for a variety of domestic purposes. And in short, as recent lineage of this group of photosynthetic microorganisms are estimated to be 5,000 with undescribed species. And as you can see from the slide, you've got examples of non-filamentous and non-flagellated algae that belongs to the green microalgae or Chlorophycota. And in our laboratory, we've got around 5 isolates that we look forward to study in the terms of biofuel or algal ketocarotenoids, on which we need to look up the different parameters, environmental parameters that we have to mimic in order to induce the microorganism, the microalgae, to produce astaxanthin. So these are the several products that we can derive from microalgae in the field of human nutrition and minimal animal nutrition, cosmetics, biofuels, aquaculture feed, and or animal feed. And recent years, there have been an increase in the amount of information on which we can utilize some genetic tools in the field of bioinformatics, transformation vectors, promoters, mutagenesis, which use as a sustainable platform for the manufacturing of several products, as we have enumerated in the slide. Furthermore, just to give you a brief background, some already commercialized products we can derive from algal biotechnology are those colorful textile dye, which is used in our, fabric. Then you've got, of course, the biofuel, which has a long history already, but 5 years or more ago, algal biofuel was used by Air New Zealand to fly using the algal fuel as an addition to the bunker fuel that they have used in Air New Zealand. Then similarly, there have been several products on which you utilize microalgae to produce meat, like the one especially from spirulina, chlorella, and the source of astaxanthin, which is used in several countries like Israel and the USA. And currently, there have been several photobioreactors which is utilized for the culture of microalgae. And this is a closed system on which they have to minimize contamination from the environment. Moving forward, there are several research projects on which microalgae is utilized. For example, this building on the left is the one which was constructed in 2013 and is already powered by microalgae, and similarly this pavilion which is powered by microalgae. And of course, we're looking forward that the green algae solutions company will succeed to produce electrical lamps out of green microalgae, on which the energy we generate, being a photosynthetic organism, they can capture the energy from the sun utilizing their photosynthetic tool. But going back to the microalgae, these are some of the several market prices of microalgal pigments. For example, we've got Haemotrichoccus, Dunaliella here, and Sendesmos species, as well as floral species. If you look at the figure, the table here, the beta-carotene, which is produced mostly from Dunaliella, which is a marine species, is about 3,000 per kilogram of US dollar per kilogram. And if you look at astaxanthin, it's about 2,500 per kilo. And of course, the phycobiliproteins, which is quite a staggering to 25,000 US dollar per gram. It is because it has several utilization in the field of biomedical field, like immunological marker in some drugs, as well as food utilization. So if you compare astaxanthin here with all other pigments derived from microalgae, like lycopene and beta-carotene, zeaxanthin, astaxanthin is the most effective against lipoperoxidation in membrane models, as you can see in the graph. And as a strong antioxidant, this is the mechanism in which astaxanthin can play its role in our cellular mechanism. For example, if this astaxanthin is absent in the cell membrane is attacked by sandali lang, hindi ko makuha yung cursor ko, hindi ko magamit. Is attacked by free radicals. And if you can see in the 1B, this leads to the activation on the NF-kB gene, which is responsible for the inflammation of activation of the NF-kB leads to the cascade of, when activated, cascade to the genes which are involved in inflammation. But on the other hand, if astaxanthin is present in the cell membrane, as you can see, acting like bridge because it has both the hydrophobic and the hydrophilic ends, the free radicals here, as you can see, into A, are getting quenched. And once they are quenched, they cannot activate the NF-kB gene that suppresses the inflammatory genes to proceed. This is because rather than donating an electron, your astaxanthin stabilizes the cell membrane like a bridge. Its polar end spans the cell membrane, increasing its rigidity, and similarly provide mechanical strength. And this would allow your astaxanthin to neutralize your singlet and triplet oxygen generated by the UVA and the UVB radiation, and then binds to the lipoprotein. So these are just some of the potential molecular mechanisms involved in astaxanthin for geroprotective effects. But in our biogenome project, whole genome sequencing will be utilized using the RBCL from several microalgal species that, when it comes to its function as primarily it's the CERT1, which is CERT1 gene, which is the most conserved mammalian nicotinamide adenine dinucleotide or NAD dependent histone deacetylase, which is attacked. And in these are proteins which are that regulate numerous cellular functions, including those aging, inflammation, detoxification, and stress resistance, fat and glucose metabolism, as well as our circadian rhythm. Moving forward, astaxanthin from microalgae is derived carotene as a gateway, coming from the central metabolism, which is for enzymatic activities before the production of beta-carotene. From beta-carotene, which is the gateway, in microalgae, we've got two enzymes which are directly involved, the CRTW, or the beta-carotene catalase, and the CRTZ, which is beta-carotene hydroxylase. So, add nyo lang ang OH at O at both ends to make the bridge stronger in order to produce your astaxanthin here. So its presence as microalgae is as illustrated in this slide, comparing the three microalgal pigments. We've got more beta-carotene in Daliella. We have more astaxanthin in Hematococcus. There is more lutein in Synedesmos almirensis. Although in our own species, we look forward that is endemic to the Philippines, which is Synedesmos obliquus. We just don't know we want to find out if it's lutein or astaxanthin present, because we just left it over the shelf and it produced an astaxanthin like pigment over time. So in essence, this is an illustration of how astaxanthin are produced in several countries, especially in Japan and in Australia. So they've got several raceways and they've got several globular tubes in order to prevent environmental contamination. So our program here, microalgal genomics structure and characterization, was submitted to PCHRD, but unfortunately, I don't know, because there's only one project which was evaluated. So we're looking forward that if PCHRD do not want to pursue this one, hopefully Picard or PCHRD can help us on this project. Thank you for listening.
Maraming salamat, Dr. Monotilla, and we would like to present the certificate of appreciation for you as well. Okay. Before we go on our quick 5-minute break, we would like to remind our participants to post their questions using our Q&A tab, and it's located at the bottom part if you're using your desktop on your screen. For mobile users, the Q&A button may be found on the upper right corner pag naka-landscape view po kayo. We have been seeing the questions in our chat room. If you can migrate them to our Q&A tab, that will be very much appreciated. Also, I would like to mention for those who are interested to collaborate and get in touch with our speakers today for their research, you can always contact our point person in the Biodiversity, Ethnicity, and Forensics program, Mr. Enrique B. Fortajada. The email address is ebfortajada1@up.edu.ph. And for more information about PGC and its services, always visit pgc.up.edu.ph or email pgc@up.edu.ph. Right now, it's 10:22. We will have a break, and hopefully everybody can return to our Zoom webinar by 10:30. Thank you so much. Welcome back, everybody. I hope you enjoyed your break. You drank your water, your coffee. And before we proceed, we would just like to reiterate that an e-certificate will be given after this webinar. Once you have accomplished our feedback form, it will take about 1 week after the webinar for you to receive your e-certificate. Right now, we have how many participants? 400 plus participants. That's great. And now, before we proceed, welcome again to Focusing on Philippine Biodiversity, Introduction to the Philippine BioGenome Project. Our next speaker is Dr. Robert S. Ginoo II from the Department of Biology in Diliman University. Dr. Ginoo received his PhD in biology at the University of San Carlos Technological Center in Cebu. And his research interests include fish systematics, taxonomy, and DNA barcoding, especially of freshwater fishes. Today, his talk is all about conservation management initiatives for the West Visayas faunal endemics at Diliman University, Centrop. Let us all welcome Dr. Ginoo.
Good morning, everyone. I'd like to share with you a presentation entitled Conservation Management Initiatives for the West Visayas Faunal Endemics at Diliman University Center for Tropical Conservation Studies. Diliman University is located in Dumaguete City, Negros Oriental, and our center is called Centrop, which is one of the leading institutions for the captive breeding of endemic and critically endangered Philippine spotted deer, Rusa alfredi, the Visayan warty pig, Sus sebifrons, Negros bleeding heart pigeon, Gallicolomba kiayi, and the Tarictic hornbill, Penelopidis panini. For the past 30 years, we at Centrop has been conducting breeding activities which resulted in a current captive bred populations of 85 Philippine spotted deer, 44 warty pigs, and 49 bleeding heart pigeons, made possible with the linkages with other breeding facilities on Negros and Panay Islands. Here is the photo of the Philippine spotted deer, Rusa alfredi, and its conservation status is placed at critically endangered, which means that the population at the wild is less than 2,500. If you look at IUCN record, it is written there that the population of the Philippine spotted deer is placed at below 1,000 mature individuals. On the right side, you will see the circle, and this is the Panay, the Negros Panay faunal region, which consists of the islands of Negros, Panay, Cebu, and Masbate. So these 4 islands are assumed to have common endemic animals, which include the Philippine spotted deer. Another animal that we had been breeding for the past 30 years is the Visayan warty pig, Sus sebifrons, known for its shaggy hair and its tusk. This is a piglet of a Visayan warty pig. We started with a few animals. For the spotted deer, we started with around 4 pairs, and at present, there are around 85 of them. For the Visayan warty pig, less than 10 was the starting population, and there are around 44 of them already. This is the Negros bleeding heart pigeon, Gallicolomba kiaye, and we started with just 4 pairs in the early 2000s, and at present there are already 49 bleeding heart pigeons in our facility. This is the Visayan hornbill, Penelopidis panini, and based on DNR, it is critically endangered, but for IUCN, it is placed at endangered status. So here's a diagram of our breeding activity for the Philippine spotted deer. And as for 2017-2018, we almost had around 100 plus, but currently the population is at 85. This is the Centrop breeding facility, which is in the municipality of Valencia. It is a 19-hectare facility, and the ones that you see here are the cages for the spotted deer. So these are activities that our staff are doing at Centrop. So to capture the spotted deer, we use a net, and then they are captured using this net. Once this net is lifted, the animal will be suspended on air, and we can place them already in this transport crate. And then upon arrival at the Centrop breeding facility, then we set them free. Now, these are the candidate reintroduction sites. One is the primary forest in Lake Balinsasayao in Sibulan, Negros Oriental. Other candidate sites include the secondary forest in Bayawan, which is in the southern part of Negros Oriental, and also in Santa Catalina, which is also located in the southern part of Negros Oriental. So this is the challenge for us, for the entire Philippines, particularly when we talk about Philippine biodiversity. The stock of Philippine endemics at Diliman is one of the largest captive bred stock globally. And this has led wildlife experts from local academic institutions and international organizations to recommend implementing a reintroduction program for these endemic species back to selected habitats in the West Visayan faunal region. But that is the challenge. When we are to reintroduce them, we need the full complement of the genetic profile of these organisms. We need to know if they are fit to be reintroduced. And so how do we achieve this? So the program that we are presenting to funding agencies as well as research collaborators such as UP and the PGC is that we propose a whole genomic profiling of the key endemic and threatened species in the Negros Panay faunal region. So this project seeks to sequence the whole genomes of 4 Negros endemic as well as critically endangered fauna. And as I mentioned, these are the Philippine spotted deer, the Visayan warty pig, the Negros bleeding heart pigeon, and the Tarictic hornbill. And what is the aim? Our aim really is to improve their conservation as well as their management. So how do we do this? How can we achieve this? So number one, we aim to sequence the whole genomes of these 4 endemic fauna of Negros. Second, to produce the DNA barcodes for these 4 endemics. So these are, the 1 and 2 are more or less, we can consider them as low-lying aims. But number 3 and number 4 are quite critical on how we manage our breeding program here at Diliman University. So the third is, we would like to develop gene markers for the Philippine spotted deer as our basis for their heterozygosity index. we need to know how many of them are fit to be reintroduced. We cannot just reintroduce them and leave them in the wild, not knowing their genetic makeup. So in captive breeding program, this is really something that is an SOP. Number 4, we've been breeding these endemics for the last 3 decades. And so there is a need to amend our existing breeding programs as well as our management for these endemics. So we need to check, are we doing the right thing in terms of how the genetic makeup of these animals are concerned. So we need to know their genetic profile using whole genome sequence. Along with this program, we have a side project, which is, again, this is a low-lying objective, and we need funding agencies as well as research collaborators to help us. So this is the cross-species amplification of microsatellite markers between the Japanese Sika deer and our Philippine spotted deer. Since the spotted deer is endemic to the Philippines and is one of the most endangered deer in the world, and at the moment, there are no specific markers yet to date for the Philippine spotted deer. So this side project aims to develop these markers through cross-species amplification with the closest deer that we can identify, which is the Sika deer. But again, there is no replacement for whole genome sequencing, which is really what I'm trying to present here. The need for whole genome sequencing of our the Negros Panay faunal endemics. So with that, I'd like to just present what are the genomic studies that are done at Diliman. So we started in 2015, and some of the studies usually conducted by graduate students include the partial express EST library of the red alga Eucuma. We also have comparison of microsatellite between 2 populations of Tridacna crozea, or the giant clam, between Indonesia and the Philippines. Another is the study on genetic diversity and discovery of a putative cryptic species within a valued crab fishery, which is the blue swimming crab. And then we also have applications of genomics in relation to the marine conservation or the marine protected areas, which our university is an advocate, the MPAs. So in these 2 papers, we tried to look into the parentage analysis of fishes coming from the MPAs and the non-MPA areas. And the findings indicate that there is really a connection between MPAs and fishes that are found in non-MPA areas. We also do some DNA barcoding, like the barcoding of Philippine mullets using CO1 gene, and also the barcoding of the Philippine freshwater anguillid eels using CO1 marker. So here are some of the results that we'd like to just present to the audience. And then this is for the freshwater eels. And some collaborations with institutions who helped us, or which helped us building capacities in terms of genomic studies. And I'd just like to highlight, this is the first DNA barcode that we did coming from the fish Oxys tazard, or the frigate tuna, locally known as ihalason, which is abundant all year round in Negros Island. We're also coordinating with other partners like the Philippine Spire, headed by universities and schools in the US. Primarily, it looks into the omics and bioinformatic aspect. So in closing, I'd like to share this idea from Gandhi. In sustaining biodiversity, and that's my idea, we need speedy action, but also correct action, because speed is irrelevant if you are going in the wrong direction. Thank you very much for listening.
Maraming salamat, Dr. Ginoo. Please stay for your certificate of appreciation. Marami po sigurong, ako po, personally natuwa sa presentation nyo dahil parang it's nice to know that Diliman University has a lot in its pipeline pagdating po sa conservation and yung DNA barcoding. Of course, one of the reasons why we have this webinar today is to add more collaborators kasi parang nakikita ko po yung mga questions doon sa panelist. Marami pong kailangan na collaborators and mga warm bodies para po sa sobrang rich ng flora and fauna ng Pilipinas, kailangan marami pong gagalaw talaga. Maraming salamat, sir.
Salamat din.
Okay. As we move to our next speaker, I would like to introduce Dr. Carmelo S Del Castillo, an assistant professor in UP Visayas, where he teaches advanced ecology, genetics, fish health, among others. He received his PhD in Marine Sciences at the United Graduate School of Agricultural Sciences, Kagoshima University in Japan. His research interests include aquatic microbiology and biotechnology, marine wildlife conservation studies, and fish immunology. Today, he will share his wide research experience in fisheries in his talk entitled Aquatic Biodiversity in the Philippines, Prospects and Challenges. Let us all welcome Dr. Del Castillo. You may now share your screen, sir.
Good morning, everyone. Can you see my screen?
Yes, sir.
Okay. So today I'm going to be talking about aquatic biodiversity specifically, and then the prospects and challenges. I guess several people already talked about the potential of biodiversity in the Philippines, the rich biodiversity. Philippines is one of the 17, sometimes 18 biodiversity hotspots in the world. And in marine biodiversity, it's one of the highest. Okay. I like this poster very much. I'm just sometimes the data presented may be out of date, but it's still done beautifully. Present here, the Philippines is one of the richest marine biodiversity in the world. The Philippines is situated in what we call the Coral Triangle, which is a hotspot of biodiversity. Sometimes it's considered that the Philippines is in the center of this Coral Triangle, and some other countries that are part of the Coral Triangle are Indonesia, Papua New Guinea, the Solomon Islands, parts of Malaysia, and of course, if we really think about it, parts of the western part of the Philippines, which is a contested area. How rich is this Coral Triangle? It's known to contain 468 corals, about 1,000 reef-associated fish species, 648 mollusk species, seagrass, about 19 species, 820 macroalgae, 3,000 fish species, about 731 of these being commercially important. These are only the species that have been identified as of now. For sure, there are a lot more that are not yet been discovered, and there's potential for much more that can be discovered in the future. From a list of the number of species as identified from the fish-based database, Philippines is about number 6 of identified species, which one of the most in the world. And among this, it also contains, sorry, I could not see this slide as I see it. It's being covered by my face. Also contains about 91 of this fish species are threatened. So what are the prospects knowing all of this? Okay. We did a study. This is under review right now. We have submitted this for publication. It's still under review. of fish species, barcoded fish species in Panay Island only. And we were able to come up with this number of species. Most of this are caught in from the markets. We cover the species from the markets. We were able to identify this, how many hundreds of species from Panay Island alone. This is a long list. I will not go into too much detail about this because it is not really a part of my presentation. But it is an interesting way to portray the rich diversity of fishes in the Philippines. From a fish base, from a personal communication, I got this list, and of endemic species in the Philippines. There are hundreds. This one is only a list of the commercially important species. Okay, so there are about, I think, 17 here. I could not see my notes, unfortunately. I think there are, if I'm not mistaken, 127 or 217 species, I think, that were listed as endemic. And then about this 17 are commercially important. Now, most of this, these are endemic in the Philippines, but sorry for the sound. I think you could hear the, it's election time. I'm pretty sure you could hear the music from outside. Now, so most of these species do not have a genome sequence yet, but I will show you later the most interesting species. There's a problem with most of them. Now, so far we've only been talking about fishes. Okay, those lists are just fishes, bony fishes specifically. And if we look at the tree of life, fishes are just a tiny part of the totality. If you put it into the aquatic ecosystem, even then, fishes are just maybe the most obvious part of the aquatic ecosystem, but it's not the only organisms that are available in the aquatic ecosystem. If you look at here, if we zoom in, this is just a small part. And if we look at this circle, actually, a lot of that are not those species that are not there anymore, except for some very unique, very few species from the colectants and the lungfishes, for example. So most of that is the branch of the bony fishes. This is what we have been presenting until now. Aside from that, just from sea life base, which again, I'm sure this is not yet complete, but this is from what is listed in sea life base. There are 113 non-fish vertebrates that are native to the Philippines. There are 1,249 crustaceans, 4,000 mollusks, 587 echinoderms, 99 jellies, 745 reef-building corals, 197 sponges, 178 marine plants. Again, these are just in the marine system. Why are these important? For example, this sponge, the Halosynthia roritzii, which I did some research work several years ago. Just a very short browsing about current studies on this came up with these two studies, one in 2020 and another one in 2022. Okay, so very, very current. And these Halosynthia roritzii have products which have medical concern. So in the Philippines, there are hundreds of other sponges that may also have this potential but have not been studied. Now, there's also mangroves. Aside from marine plants that were listed, mangroves are not necessarily marine plants. Those are marine algae that were listed there. So there are also 46 out of the 70 salt-tolerant mangroves. 46 of those are found in the Philippines. And these mangroves may also have some medicinal properties. So aside from the desire to protect the environment, a whole genome sequence may also have other practical interests. Okay, for example, in helping look for potential agents against cancer, against some diseases. Knowing the genome of an organism can help in finding those things. Now, until now, those species that we listed, they are just a small part of the totality of organisms. As Sir Fontanellia, I think, mentioned earlier, that aside from plants and animals, there are also bacteria, there are also fungi, there are even viruses. All of these have genomes that are of interest to learn. Now, this is the circle of life as available in the Open Tree of Life and as published in Scientific American. In here, the what's this? The arthropods are in this area here, and then metazoans, vertebrates would be somewhere here, I think. It's a very small branch. Now, even this is actually a bit cladocentric already. This representation of the totality of life is already somewhat cladocentric. I don't know how to undo the annotation now. Clear all drawings. Okay, so I was able to undo the annotation. Sorry about that. Now, this is another representation of the tree of life, which is based on diversity. Okay. Now, if you look at this tree of life, if you look here at this area, this is a large, huge branch. And then this is branched here to another clade. If you look at that clade only, put that inside a square and look at that part only, you will see here that mammals form only a very tiny part of that clade. Okay, fishes, so this branch here would be chordates. Okay, this will be a branch for the chordata. This will form a tiny branch. All animals, all animalia, including non-chordates, they would form a tiny branch of the biodiversity of all total life. Eukaryotes is a tiny branch. Eukaryotes would include worms, plants, all the fungi. This will form a very tiny branch of this clade. A huge branch would be just simple bacteria. And those by themselves are also part of the totality of life. Whole genome sequence of those would also be interesting. Now, there are certain challenges available. I'm going to share a conversation. This is a real conversation between me and one of our consortium members. So I asked him, okay, I'm redacting the names and then some of the details because I'm still working on some of this. So I'm only sharing some of the details here. So I asked my coworker, hey, gawa tayo ng proposal para dun sa Earth BioGenome project. This includes all the misspellings as you type. Yes, sir, I'm thinking about the sequence of humpback groper. Oh, available na yan. Okay, 2020 yan, published. But actually, I was actually mistaken. This is still BioRibX, so it is still under review. So this is not published 2020. It is still under review for 2022. Okay. So about coconut crab, sir, Napoleon Ras. So I asked him, Virgus latro? Yes, sir. Ayan, available rin yan. Published na yan. Okay, on chromosomal level. Okay, how about, sir, yung kwan? So I showed here ang Virgus latro and then the humphead ras. Okay, and Napoleon Ras and humphead ras. So Chelenos undulatus also published. How about Selenota ananias and Actinopyga? Okay, ananas available rin yan, published rin yan. How about ang Actinopyga? Published rin yan. Now, what is interesting is to see, as I'm going to show in the next slide, when these genomes have been published. Okay, so for the humpback groper, as I said, this is still with RibX 2022, so it's probably going to be published 2022. The Bigrus latro, the nuclear genome, has been published. The manuscript is still under peer review. The Chilenos undulatus, the Napoleon Ras. Okay, this was published 2021 of October. So it was just available just last year. Prickly redfish, Silonata ananas, published 2021, about July, available online. So it must have been published just after July 2021. The Actinoptiga, published 2022, I think. Yes, 27 January 2020. Okay, so all of these very recent, and also interesting. I included there the author's information for for interest. None of those are in the Philippines. Now, another problem is, for example, the genome of our Philippine national fish, Chanos, Mangroves. This has been available also been available since around 2019. Okay, also a very recent assembly. This is the biggest aquaculture in value and in volume among fish. In volume, seaweeds is number one, but the value of seaweeds is very low. But it is the Chanos, the Bangros, is the biggest in value and also in volume among the fishes. And I'm just showing the picture to you guys because it's almost lunchtime. I hope you're going to be eating some aquaculture product later for lunch. But this assembly is very recent. A few years ago, we were making a proposal on a Bangrus, and then this one has not yet been openly published. Now the assembly has been published since 2019. Again, this one is being done by I think a US group. And then another problem. So despite the words to the contrary of interest in on proposals on whole genome sequences, we just submitted just March 31, just last month, a proposal on bioprospecting a native species. It's a very interesting native species. It's a symbol of Philippines, actually. We submitted a proposal for the bioprospecting of this species for medicinal and pharmacological genes. So in order to do that, we do a whole genome sequence, and as you can see, it was disapproved. Okay, so these are still some of the challenges, despite words to the contrary. Seems the funding agencies are still not very open to these kinds of proposals. So in summary, what are the prospects in aquatic biodiversity in the Philippines? It's huge. The Philippines is one of the most biodiverse aquatic environments. And this is not just limited to fishes. This can be all other, as I said, sea squirts, jellies, algae, and of course that's not even counting the microbiota, which is again another very huge potential in itself. And the capability to do whole genome sequence is now here. The expertise is here. The equipment is now here. But the challenges that are facing us right now is, first, other people are doing it for us. So we're running out of time. The problem, one of the problem in compared to land-based animals, in order for a foreign entity to sequence Visayan spotted deer, for example, from Dr. Ginoo, they have to come here. They have to get a sample from Negros or Panay and go back to their country to sequence it. The problem is fishes, they're running around in Sulu Sea, they're running around in Palawan, they're running around in West Philippine Sea. And in West Philippine Sea, there are people who can easily go there and sample and do the sequencing for us. Okay? So if we don't act on this right now, we'll run out of fishes to sample, because somebody else will be doing it for us. Someone who has a lot of capability and who has projects that are mass sequencing of genomes. Okay, and another problem is, of course, still the interest of funding agencies. Hopefully, this will change as as Secretary Peña just mentioned, that there's going to be interest in this. Hopefully, this will translate into reality soon, that the interest will come in the real funding. Now there is funding for the PGC. The PGC is there. Who's going to use the PGC if there's no funding for other works to utilize the PGC? Okay, so these are challenges, and hope that these challenges, we can overcome these challenges, and we will able to move forward with all the potential that is available to us here. From our part in University of the Philippines Visayas and the Philippine Genome Center Visayas, we are always welcoming of other agencies, other SUCs that can collaborate or work with us as a consortium, or just work together with us. The proposal that I showed you was not my personal proposal, but it was the proposal of a consortium, one of our members of our consortium. We're also working together with other universities. Hopefully, some works are going to be realized soon, and I hope it will be done soon. So, as Dr. Ongria has mentioned, we're always looking to work together with other people. Thank you for your time.
Maraming salamat, Dr. Del Castillo. Very impassioned, and ang galing ng resibo ng talk mo, sir, pati yung text na include natin. Very, very informative. And to move, before we move on to the next speaker, we would like to present the certificate of appreciation for you as well.
Thank you.
For more talks in the future, lagi ka naman nandyan, sir, for PGC. For our next speaker, let me introduce Dr. Zenaida Bawanan. a professor in the Department of Biology in UP Baguio. Well represented ng mga UP campuses ngayon where she teaches biology, invertebrate zoology, ecology, and she received her PhD in biology at UP Diliman and was a Fulbright postdoc fellow at the University of Hawaii at Manoa. She currently heads the Biodiversity Resource Management Program at the Cordillera Studies Center in UP Baguio. She is actively conducting relevant ecological and conservation research with local and international collaborators. Please welcome Dr. Bawanan for her talk entitled Tapping Non-Marine Bivalves and Snails for Sustainable Utilization and Consumption in Luzon, Philippines.
Thank you, Ms. Cheryl. May I now share my presentation? Hello, good morning, good noon, everyone. Thank you for staying with us and thank you for this opportunity. What I'll be discussing for this morning is a bisocol prospecting, and this expands to tapping non-marine bivalves and snails for sustainable utilization and consumption in Luzon, Philippines. Bisocol is commonly known as, is a common name, Ilocano term actually, and also a Pangasinan term for kuhol. And may I just give you some interesting facts about the mollusks. So it is the second largest group of animals next to arthropods, or the phylum of the insects, with about 200,000 living species, of which 31% are found in the Philippines. Under the phylum Mollusca, the gastropods and the bivalves are the two largest classes, and 50% of the reported 1,294 Philippine land snails are actually found in Luzon. And we cannot discount the importance of the mollusks. In terms of their biological significance, they play an important role in the food chain or in the food web. And they're also important decomposers. Thereby, they return the nutrients to the soil to become available for the plant utilization. And because they leave a hard shell, majority of the mollusks would leave a hard shell, then they are also an important model for biogeographic and evolutionary studies. Because the mollusks, particularly the snails and the bivalves, are slow-moving. Kaya nga sinabi, di ba, pag mabagal ka, sabihin nila, para kang suso or para kang snail. Okay? And so they play an important role as indicators of a healthy ecosystem. The more diverse snails and bivalves there are in an ecosystem means that the ecosystem is still healthy. In terms of their significance to human, most of the introduced species here in the country has become an agricultural pest. Examples of this, or notorious to this, is the Pumasea canaliculata. There are already several reports about the impact of its introduction to the country. And we also have the Lysacatina fulica, or also known as the giant African snail, that was also introduced during World War II. And recently here in the Cordillera, even here in Baguio City, there is already the spreading of these Macroclamis chelantenensis. They're found in the parks here in the Cordillera, and even in the sayote plantations. And they are also highly prized collections and crafts. Example of this is the Conus Gloria Maris, or the cone of the Glory of the Sea cone. And in 1970s, this was auctioned to as high as $2,000. And there are also important crafts. If you go to Palawan, you can also see a lot of the shells that are being sold in the market and elsewhere here in the country. Now, the pearl farming, particularly of Pinctada maxima, which is a sea oyster, has contributed to the economy of the local people and also here in the Philippines. Now, The snails are also important intermediate hosts of parasites. Example of this is the Oncomelania hopensis quadrasi, which is an intermediate host of the parasite Schistosoma japonicum that leads to the disease schistosomiasis. And we have actually used environmental DNA from the soil in order to detect the spread of this Oncomelania hopensis, which is an intermediate host of this important parasite. And they're also used as our ornaments and tools. In fact, in certain place here in the country, like in Batanes, yung mga na-excavate nila ng mga shells also show us that even in the old times, the shells are already important source of ornaments and tools, and also used as food by the local people. You can see here my personal items, and this one is a cone snail that was used as a bait for octopus, and this one is a shell of a terebra that is used as a handle for a magnifying glass. And the set of jewelries that I'm wearing now are actually pearl from Palawan. And most importantly, the snails are an important source of the natural products. Of course, you are most familiar with the cone snails as one of the richest source of bioactive peptides. And the Conus magus has become developed into biologically based medicine or analgesic called the Prialt. And other than that, the cone snails are also potential source of molluscicides and also insecticides and antimicrobial agents. So there's a lot of potential. But did you know that even the land snails can also become a possible source of cosmeceutical products? The Lysacatina fulica here, or the giant African snail with its yellow eggs, has become a well-studied source of natural products. And as I've mentioned earlier, this was introduced to the Philippines in 1942 by the Japanese soldiers. They brought it with them and used it as their food. However, after the war, they've left some of the snails in the wilderness, and it has become widespread. And as we know, for some introduced species, because they don't have the natural enemies to regulate their population, so once they have established their population, then they can become invasive and become pests. However, since Lysacatina fulica has been introduced to the country since 1942, they might have already been started to be naturalized in our environment and may have had ameliorated their impact as a pest. And this Lysacatina fulica is now a viable candidate for snail farming because of their large size, and they are adaptable to wide range of food and environmental conditions. Now, in 2017, I had the opportunity to go to Bangkok, Thailand, and I was with Dr. Ian Fontanellia, and we visited the Siam Snail Eco Farm. Now, the reason for our visit in Thailand was that we were hosted by Dr. Somsak Panha to organize a malacological association among Southeast Asian nations. And we will be having our first conference this year. So I hope that you can also participate in that international conference. And in the Siam Snail Eco Farm, they're particularly using a Hemiplecta distincta. So you can see here at the background of our picture, the snail as the source of their snail-based products, but they're also rearing some Lysacatina fulica. And of course, I guess you're also familiar with the South Korean hype na yung mga magagandang kutis ng mga Koreans, and nag-boom din yung skincare products in South Korea way back in 2016. And I think some of these products are now commercially available here in the country. Now, gastropods are also gastronomic delights. Particularly in Europe, merong mga canned na escargot snails, and these are some nutritional information about the helix snail meat. The land snails and bivalves has become a part of the Filipino diet even way back sa mga ninuno pa natin. And in the CHED, there too funded research that that was only finished last year. Then we were able to document some land snails in the northern Luzon. And interestingly, in Batanes, there is this snail which the Batans would call marida, and this is Hemiglypta coveriana, and they use this as a food. And even the beber, the locally known beber or the Calocochlea pluchemira, and the Helicostyla grandis in Isabela and Cagayan, known as talcoc. So these are also edible snails. And in southern Luzon, we have this bayuco or the Rhizota otahetana, which is also hunted for food. Now, preparations for these snails would actually be either as is in a form of broiling, or they also cook it as adobo, which is an used as delicacy or pulutan. And so, because of this high pressure in the collection of these endemic snails, they are also threatened in the wild. This is the list of some edible snails and bivalves that was reported in Cagayan River in 2006. Some of these thiarids and also viviparids are utilized as duck feed, which are also used as lime source for betel nut and for killing unwanted earthworms in Cordillera. Now, the accidentally introduced Crestaria plicata and species of Anodonta are already being used for freshwater mussel pearl farming here in the country. Now, in this study by PISEA in 2021, this figure shows the untapped value of the land snails in the Philippines. The meat, which is actually part of the food, is an important source for protein, and it is low in fat, kaya maganda siya para sa mga tao na nagda-diet. And it's also rich in calcium and in potassium. Now, the shells coming from the snails are also locally used as a lime for not only for betel nut, but also for neutralizing the soil. And then the gelatin recovery from the meat of the snails is still a research gap. There's not much study that is being done yet on this. And then the visceral mass, which constitute the internal organs of the snails, are usually being disposed or waste. Hindi siya masyado nagagamit. But of course, it can become an organic fertilizer if you just dispose it. Now, the eggs are also used as a white caviar kasi yung mga majority ng mga land snails produce white mass of white egg mass, and para din silang mga caviar na naproproduce din ng mga crabs. And this could also be a highly priced commodity. And the mucus, so you would know kung may dumaan dyan na snail or slug kasi may makikita kayo dyan ng mga snail track or mucus track. Now, this mucus is becoming an important source of natural product, which I have mentioned earlier. And the chitosan from the mucus of the land snails can also be used for packaging. Now, heliculture is not new in other countries. It has started in Europe for snail meat delicacy known as the escargot. And it has been used to alleviate the poverty in Nigeria since early 2000, but this is not yet a recognized aspect of livestock production in the country. But there are already booklets and books about snail farming, which we can use if we wanted to establish a snail farm here in the country. And compared to aquaculture, snail farming or heliculture has a lower environmental footprint because the idea is just to put the snails in a semi-enclosed environment or an enclosed environment, but they are still in their natural habitat. So meron na talagang mga nakatanim dyan na mga local vegetation at doon sila mag-feed. So very minimal lang yung intervention ng human. So magkakaroon lang dito ng mga intervention doon sa pag-develop ng skill in extracting the snail from the live individual. And so this can become a sustainable source of mucus which we can use to tap different natural products from the land snails. And so these are some of the research gaps that we hope to address: the need to evaluate the sustainability of the consumption and the utilization of the non-marine mollusks, because we know already so much about the marine counterparts, but not so much on the non-marine mollusks for food security. So in fact, we have a proposal with Picard, and this is on invertebrates for food security. And this will be on the edible mollusk and insects of Luzon. And therefore, the whole genome sequence will become a tool to tap the hidden treasures of our silently threatened endemic snails. And we also have a proposal with the Philippine Council for Health Research and Development, who is also sponsoring the Tuklas Lunas Consortium that is being led by UP Baguio together with Benguet State University and St. Louis University. And we have proposed research on exploring the wound healing properties of the land snail mucus that will be conjugated with the nanoparticles. And to have a sustainable source of mucus, we hope to also establish a snail farm here in Benguet that is in the land owned by the UP Baguio, which is located in Subkil. And you can see in this picture the Drycocochleas butleri, which we formerly know as the Helicostyla butleri, as an endemic land snail here in the Cordillera, which we could probably also tap for our research. And this review paper that was recently published in 2020 tells us about the gap in the molluscan genomics, that we are still way far on our road to understanding molluscan genomics, and we really look forward to address this gap in molluscan research. And with that, I'd like to thank you for listening, and I wish to acknowledge the CHED there too for funding us and in our project, and we were able to document some land snails here in northern Luzon. And I also would like to thank the Philippine Genome Center for sponsoring this and for us to be able to promote our advocacy, our research interest, and also my co-malacologists who have been my collaborators in my researches in malacology, Dr. Ian Fontanellia of UP Diliman, Dr. Emmanuel Ryan de Chavez of UP Los Baños, and Dr. Giselle Batumalake. Thank you so much.
Thank you, Dr. Bawanan. And we now present to you our certificate of appreciation for all the hard work and for your participation in this webinar. Marami pong salamat, ma'am.
Thank you din.
Okay. So, nasa huling speaker na po tayo, no? Pero hindi po yun ibig sabihin na hindi exciting. Sobrang exciting na itong bagong speaker natin. Nakapag-talk na tayo tungkol sa mga eagles, tapos may bats, tapos kanina snails. Ngayon naman, hulaan nyo kung tungkol saan yung talk natin. Very interesting siya. And right in time for lunch din. Let me introduce our last speaker for the day. It's Dr. Michael James P. Herrera. Dr. Herrera is an associate professor at the Archaeological Studies program in UP Diliman. His work has centered around the genetics of commensal organisms as a proxy for addressing research questions on Philippine prehistory. Ayan, tungkol sa Philippine prehistory. Specifically, he has been using domestic chickens for understanding prehistoric human migrations in island Southeast Asia and the Pacific. He also uses commensal rodents to study early agriculture in the region. His talk, titled Chickens Across the Sea, Hypothesis, Testing, and Conservation Using Archaeogenomics. It's a very interesting field, personally for me, because basically it's the first time for me to have this concept presented. And I would like to welcome Dr. Herrera to share his screen.
All right, okay. So thank you very much. Magandang umaga sa lahat po. So ako po si Michael, coming from the Archaeological Studies program. And this morning, I'm going to be talking about chickens. Okay. So just to show you the progression of my talk this morning. So first and foremost, I'm going to be talking about chickens and why do we need to bother about them. So it is very apparent to me that the species or the taxa mentioned earlier are quite rare. They're quite unique. They are endangered. Okay, and then we can never think about chickens being endangered because they count in billions. Okay, they practically outnumber us. Okay, but allow me today to tell you a slightly different story and why it might be important to actually help native chickens, particularly in the Philippines. Okay, so and then of course I'm going to tell you how chickens are very good storytellers. Okay, about our past. Okay, so particularly the story that they allow us to glean upon is the prehistoric human movements in the Asia Pacific region. Okay, and then I'm going to impress on you also later that the genetic heritage of chickens, okay, in the Philippines is quite unique, okay, quite worthy of protection as well. And then the last bit that I'm going to share with you guys is that how do we improve, okay, in telling the story about our past, okay, using chickens and other commensal animals. Okay, so dun papasok yung archaeogenomics. So in a practical sense, okay, so it's using genome level data, okay, but however it has a time transect to it. So it means that we're going to look at ancient DNA. All right. So honestly, half of the time when I think of chickens, I think of them as this. Okay, so I think of them as Chicken Joy, KFC. Okay, and then however, the other half, okay, I think of chicken as a very serious or a very important thing to look at, okay, for, again, understanding the past and, of course, for food security. Okay, so things like those. Okay, and then hopefully at the end of this talk, I'd be able to convince you guys that the next time you eat Chicken Joy or KFC, okay, there is heaps more behind those crispy skin. All right, so the chickens that we normally consume on a daily basis, okay, are chickens, okay, that we see in very big farms such as this. Okay, so these are the commercial chicken lines. Okay, today I will demonstrate to you that the chickens that we do find here in the Philippines are quite different from these chickens. Okay, their genetic lineages is quite unique, and then the commercial chicken lines that we have right now, okay, they belong to a very small group of genetic lineages in chickens. Okay, so it means that their low genetic diversity will have important implications, like in terms of like diseases, pandemics. Okay, and then having an idea as to where else can we find genetic diversity in chickens is actually very important in ameliorating those problems in the future. Okay, so I'm going to talk more about that in a little while. Okay, now the work that that we've been doing on chicken is rather a protracted one. So when I did my PhD, I worked on chickens, and then currently I'm still working on chickens, okay, particularly in Zamba Sulta, so that's Zamboanga, Sulu, Basilan, Tawi-Tawi. Okay, so this particular sampling picture here was done in Basilan. So we were going around, okay, and then collecting chicken feathers. So our approach is a very non-invasive approach. So we basically just pluck chicken feathers, okay, and then so that allows us that allows us to actually access the chickens from more, okay, from the owners, okay, instead of like getting blood, for instance, okay. Okay, so these are more action shots from Tawi-Tawi, okay, with Jay Rodriguez, who's also working on human genetics from the region. Okay, so and then of course we interview the owners, we get as much metadata from the chicken. Okay, all right, and then in our travels, okay, like collecting chicken feathers, okay, from across the Philippines, okay, so we've realized that they come in a great diversity just on the plumage alone. Okay, so you can see here that you've got them coming in different colors, coming in different sizes. Okay, their legs come in different color, gray or yellow. That has important meaning as well. Bakit yellow yung paa nila or gray yung paa nila? Okay, and then of course lower corner left side, that's a chick of a labuyo. All right, so here's more picture of our sampling trip. So they come in great diversity, at least in terms of plumage. Okay, now why genomic studies on chickens are important? Okay, now for my purpose, coming as an archaeologist, okay, so chickens are very important actually in allowing us to have an alternative view of how human colonization and settlement happened in island Southeast Asia and the Pacific during the past. Okay, now of course there are a lot of work done by people using human genetics. Okay, but sometimes there will still be like times wherein hindi masasagot or hindi may kailangan ng clarification, and then sometimes using commensals or animals, okay, or items taken with people during this colonization can actually help us, okay, in resolving those issues. Okay, and then of course, having a genomic knowledge of chickens coming from island Southeast Asia can allow us to actually concretely develop livestock conservation. And then of course, that will have an important impact on food security. Okay. So just very quickly, just to have a little bit of context, like how am I using chickens as a tool to understand the past? So practically the regions that I'm most interested in would be, of course, island Southeast Asia and the Pacific. And then the culture, it would be the Neolithic period, and of course, yung geological time scale, okay, somewhere during the Holocene. And I'm particularly interested, okay, with the Austronesian speaking people, because most of the hypotheses about the spread of agriculture is thought by scientists to have happened, okay, side by side with the Austronesian speaking people. And of course, in that migration or in that movement, okay, napaka-importante that we tease apart ano yung mga interactions that happened, okay, during that time. So yung cultural contact, okay, the trade that they were doing, and of course yung biological translocations. Now I say biological translocations because it's not actually just chickens that got transported across the region during prehistory. Okay, and then among one of the contentious, I suppose the two most contentious bits, okay, that people are talking about human colonization in the region is, of course, whether the cultures that we've got in here are internal development or whether it actually involved exogenous movements of people from other areas or regions. Okay, and then I think, okay, and I will argue that chickens and other commensals are actually very helpful in resolving those. All right, now so as I've mentioned earlier, a lot of researchers have been looking into the human colonization process of island Southeast Asia, and then some of those. So that's why we do have a lot of models. We do have a lot of hypotheses, okay, about those movements, and you can see that they come in varying forms here. Okay, from the most simple one, which is like panmixia, okay, where there's no what is genetic structure, to one that there's a clear phylogenetic signal. However, they have varying trajectories. So what we are really hoping to do is that once we use the chicken model, okay, using the frameworks of phylogeography and phylogenetics, okay, population genetics, and then hopefully, okay, we'd be able to like see which of these models, okay, are which of this model is best fit, okay, in comparison to the chicken data. Okay, now very quickly, let me talk about bioproxy. So basically, these are just, we know we have archaeological evidence, okay, that people making long distance travel, particularly, okay, in the Pacific and in island Southeast Asia, that they were bringing items with them from different places. Okay, now this was actually part of their survival strategy when they make long distance crossings across open seas. So kailangan nilang magbaon ng food, okay? Now sometimes they bring these food items or different taxa to eat during the travel, and sometimes to actually establish in the newfound islands. Okay, so once we've established that that they were indeed translocated in the past, and then they can actually serve. So it means that they actually share a common history with us. Okay, and then we can use them as proxies. Okay, so they're kind of like analogous to like living artifacts. Okay, so most of the time they give us an alternative story. Okay, which is interesting, and of course sometimes it gives us complementary stories. Okay, now just as a demonstration, okay, proof of concept. Okay, there's been a lot of work done. Okay, on bioproxies. Okay, and then of course particularly on piecing apart the past. Okay. All right, now in our region, in the Asia Pacific region, okay, there are 4 storytellers, okay, that we can use as a bioproxy. Okay, so of course it includes dogs. Okay, it includes the Ratus excellens, or the Pacific rats. Okay, pigs, and of course chickens. However, I will argue today that chickens are actually one of the best bioproxy that we could use, particularly in the Asia Pacific. Okay. So with those 4 organisms, or those 4 taxa, okay, current studies or recent studies about on their genetics, okay, have revealed different stories. Okay, one of the biggest story that they are telling us, okay, and then which is in contradiction to how we understood, okay, the dispersal of the Neolithic in the region, is that they did not come as a package. Okay, so where their homelands or their places of origin, okay, the trajectory that they've used, okay, and of course the timing as well is very different. So they did not come as a package. So we have to like dismantle, okay, the quote unquote the Neolithic package. So iba-iba yung nila yung stories nila. So that's one thing. And then in those studies, ang ginamit natin ay are haplotypic markers, like for instance mitochondrial control region and the Y chromosome. Okay. And then today I'm going to impress on you as well why it is important to actually to use a genome scale or a genome level data. All right. Now we've gleaned like the stories given by dogs, rats, and of course pigs. Okay, but however, we do not actually know the story until quite recently, okay, about what's going on with chickens in island Southeast Asia. Okay, so there's a gap in the research until recently. However, okay, in the Pacific, okay, we know we excavate them, and then and the ancient DNA sequences, modern DNA sequences have been produced on chickens. And then all of those information were used to actually reconstruct the story in the Pacific. But the obvious region adjacent to the Pacific where chickens, okay, probably most likely came from, we've got, there are still a lot of things that are not resolved. Okay, but before that, before going into that story, okay, now why chickens? Okay, now for obvious reason, okay, chickens can serve as a living larder, okay, as I've mentioned earlier, when they move from island to island, okay, and then they've got a very short generation time, okay, so it means that they are easy to to paramihin. Okay, they're quite easy to keep as well. Then of course, for obvious reason, because I imagine that people in the past would have used small boats to cross the oceans, to cross the Pacific. So they are highly transportable due to their size. And then, of course, culturally speaking, okay, cockfighting is a very big thing. Okay, not only in island Southeast Asia, but in the Pacific and even in South America as well. And of course, they can serve as a very good indicator of social status as well. And I think if you're going to look at people who keep cockfighters, okay, so we know that they are highly priced, quite expensive. Okay, so, and then of course they can serve as a spiritual accoutrement. Okay, and then of course for obvious reason, nakita natin si Isabel dito, she's plucked a lot of chicken feathers to make her costume. Okay, however, one of the reasons why chickens are quite important as a proxy, okay, for understanding human movements in island environments is that chickens are unable to sustain long distance flight. Okay, so if we see them archaeologically, okay, like say for instance in the Pacific, okay, it means that they got there by virtue of human translocation, by virtue of human agency. Okay, and then there's no alternative to that because biologically, chickens are really unable to like sustain long distance flight. So it means that whatever story, okay, that we can gather from them, we do not have to worry about that issue about them getting there on their own. Okay, similarly with pigs, medyo hindi kasi kaya ng pigs ng island, they can swim between islands, okay, and then there are like it's been demonstrated before, okay, so that's why its value as an organism for reconstructing past movements in the region is rather low in comparison to chickens. And then similarly with Ratus excellens, yung motivation, some scientists would argue that Ratus excellens is not a good indicator for translocation because they can hop on the boats without people knowing. So yung intention, okay, of movement is quite needs to be proven. Okay, but we know that certain cultures in the Pacific, they do eat Pacific rats. Okay, now another reason why it is important to look at rats, because in comparison to the other domesticated, okay, other commensals, okay, so chickens actually show a very high level of phylogeographic structure. Okay, so it means that there's a clear signal, okay, that we can see, that we can potentially see, okay, in terms of like trajectory, where they come from, okay. So makikita natin, we can map it out, okay, on across the landscape. So one important implication to that is that they could potentially provide as a means to search for the original founding population of chickens in the Pacific. Now, even in the Philippines, okay, we still have, at least I believe, okay, that the story about the labuyo, which some researchers would argue that they arrived in the Philippines, okay, at some point in the past as domestic animals that eventually went feral. Okay, but considering the work that I've done on chickens, I think I'm beginning to think that there could be an alternative to that. Okay, so however, we need more work to do to actually prove that. What I'm trying to say is that chicken, I think that the labuyo chicken could potentially be endemic, okay, to the Philippines, in the Philippines. Okay, so in the work that we've done, okay, so we've sampled chicken feathers, okay, we've sampled chicken bones, we've sampled toe pads, okay, from across mainland Southeast Asia, island Southeast Asia, and the Pacific. So it took us a while, okay, to build our sample set, okay, and then of course you can imagine, okay, the amount of letters that you need to send, okay, to people to actually allow us to access their museum collections, okay, and then their archaeological collections as well. Okay, now with the work that we've done on chickens, okay, so we know that globally, okay, chickens have globally the diversity of chickens would fall in any of these letters here, okay, in any of this group. So we know that chickens, at least in island Southeast Asia and Oceania, they actually belong to either D or E, and then they come at a very low frequency for other haplogroups. However, this particular work, okay, was done back in 2014, okay, but right now, okay, we know that we know that chickens in island Southeast Asia, although they belong to haplogroup D, D actually contains a very big amount of diversity itself, which developed locally in the Philippines. Okay, so that's something very interesting. So we can remove, okay, we can remove A, B, C, okay, and then the other letters, okay, in our next slides, because we now know that like 90%, 90+% of them would fall under D, and then at a very small frequency, letter E. Okay. However, so although we know that we've got haplogroup E and haplogroup D, okay, so there is an issue with haplogroup E. Okay, so yung haplogroup E, it is actually the lineages, okay, that we see in most of our commercial chicken lines. Okay, now the problem with that is that they do not actually show any phylogenetic signal. So as demonstrated in this MDS here, okay, so a global scenario that we have here is that you cannot actually detect a level of any structure or phylogenetic signal. Okay, so which becomes very hard, okay, in terms of like extrapolating this to understand the past, because it's panmictic. Okay, now, however, okay, if we look at haplogroup D, okay, so there's signal, okay, and then there's structure to it. So a global data set, okay, would tell us that there is a split between continental populations, save for some, because we've got Cebu, Batanes, okay, on the other side of the graph, and then we've also got the insular population. So more or less, you can split them that way. Okay. However, looking just at the insular population, which you can see the figure, the lower right side. Okay, you can see that you've got Philippine chickens, oceanic chickens, and the Indonesian chickens, Indonesian populations. You can see that quite quickly that Philippine populations would tend to like associate more with Pacific chickens. All right, now archaeological chickens. So there is actually a bit of an issue with archaeological chickens because we don't seem to like dig them here in the Philippines. Okay. However, in the Pacific, okay, with similar environmental milieu, okay, to the Philippines, tropical climate, high humidity, stuff like those, okay, we do actually encounter them there, okay, but not in the Philippines. So some of the chickens, okay, excavated in the Pacific, okay, like say for instance, coming from Easter Island, generated a signal, okay, they belong to haplogroup D. However, they belong to a very small set of haplotype, which contains 4 SNPs, okay, that can only be found in the Pacific. Okay, and then so that became a point for us, okay, to actually allow us to help find the homeland of the Pacific or the Polynesian chickens, because we now know of that 4 Pacific SNP. We call it the ancestral Polynesian chicken or SNPs. Okay, so it's really a matter of looking westward, because we know that chickens are not endemic in the Americas, so we look westward, so in island Southeast Asia. So we did that survey, okay, and then when we collected like heaps and heaps of chickens from Philippines and Indonesia, okay, so we've realized that yung haplogroup D, particularly those lineages or haplotypes that contains Polynesian D, okay, we tend to find them only in the Philippines. Okay, so in this particular data set, we have approximately around 800 chickens coming from Indonesia, and then we did not see that signal there. Okay, so that's to me, that's something very exciting because it points to the origin of the Polynesian chickens. Okay, now what we want to do now is that we want to test that hypothesis or that story, okay, now using a bigger data set. So in here, okay, we've used whole mitochondrial genomes. So because the producing these genomes is quite expensive, so we do not really have all of the, we cannot afford to actually have all of the samples that we've got here sequenced for whole mitochondrial genomes. It's just a small subset. However, very quickly, you can see that there's 3 at least groupings here. So you've got the Austronesian chicken, island Southeast Asia diversity, and of course coming from Asia. And then you will see that the Polynesian chickens, okay, they seem to like jut out from a diversity coming from the Philippines. Okay, so that's something very interesting, okay, in terms of like looking at this story using the whole mitochondrial genome. Okay, now of course, we want to demonstrate the observation using different statistical approaches. So we've used PCA, we've used maximum likelihood here, and then the groupings observed in the previous MJ network, okay, is still telling us the same story. So I'm quite happy about that. And then of course, here is a Bayesian inference of that same data, not same data set, this is control region, but generally what you will see is that Pacific chickens, okay, seems to be nested, okay, within Philippine chickens. All right, now why go genomics or archaeogenomics? Okay, so yes, we can use modern specimens, okay, to tell us about the past. Okay, we can extrapolate or reconstruct the past using them. However, nothing will beat the value, okay, if you have a time transect to that data. Okay, so what I'm saying is that if you have ancient DNA, or if you can access it, and then that's a direct it will directly tell us the story of the past, okay, instead of having to extrapolate it with modern data. However, there are a lot of challenges if you do that. Okay, so for obvious reason, it's quite expensive to do it, and of course, again, conservation issues, particularly for ancient DNA coming from the tropics. However, it can be done. Okay, so I promise you that. Okay, so we've reconstructed, or we've done our storytelling using mitochondrial control region. Okay, we've used whole mitochondrial genomes. Now, I am here right now telling the group that we should also actually test this hypothesis using whole genomic approaches, okay, because for various reasons, okay. So one is that the resolution, okay, that we get, okay, if we use whole genome sequencing is of course much better. Okay, I really believe in that. Okay, and that it's been demonstrated in a lot of papers. Okay, and then of course you've got a temporal distribution, okay, of the different taxa that we are interested in, not just a spatial one. Okay, so again, a time transect. Okay, and then of course it provides a useful framework for researchers and managers alike in their effort to protect and preserve biodiversity. Okay, now with whole genome, so we know that if we have a sample and we get the genetic data from it, so we can estimate phylogenetic trees, we can do multivariate, so we can do all of these things using modern genetic data. However, okay, using a genome scale approach and having ancient DNA produced for the same specimen, okay, it will actually improve the resolution of our data. So like say for instance for demographic modeling, okay, so you will, at least for me as an archaeologist, I am interested as to how the genetic diversity changed through time, okay, and then under the context of different climatic milieus or different cultures, okay, and then so it's interesting to have that layer of data as well. So that's why I'm pushing for the group to actually look at ancient DNA as well. Okay, now in our study, okay, so this is a bit where I tell you why, okay, the labuyo or the red jungle fowls, okay, are quite interesting, and then we should really look at it closely. So we've estimated in here a PCA using a 600,000 SNP data, chickens coming from Asia, the Philippines, and Oceania. And then one of the bits that you can see in here is that our red jungle fowl, okay, they form their unique group in there, okay, and then that makes me hopeful. So in terms of like understanding admixture, maybe potentially we still have red jungle fowls, okay, that are more or less in a sense genetically quote unquote pure. Okay, so that's very good. And then of course there's a diversity that's unique to Philippine domestic chickens as well, which is different from mainland Asian chickens. Okay, so it's the same data set, okay, but presented, okay, differently. So you've got, again, a PCA here, okay, so you can see the Philippine red jungle fowl, and then of course the Batanes chicken kind of like having its own group in there, the Pacific as well. Okay, but however, in this admixture analysis, okay, so it's interesting to see that yung red jungle fowls coming from the Philippines, they tend to have like their ancestry seems to show us that medyo isa lang siya. Okay, so I think that's interesting. Okay, so at least at this stage, okay, we can say that potentially the admixture event, okay, experienced by the red jungle fowl is rather low. Okay, but I don't know because I only have a handful of samples thus far. Okay, so together with Ma'am Cora, so we are like working out, okay, more. We're like we are like working more on chickens to actually to tease apart the reasons behind what we are observing in our data set now. All right, so the challenges, okay, and of course, and it has been mentioned by previous speakers earlier, okay, so there's actually a gap between academia and policymakers, okay, and then there's a lot of reasons behind that, and then hopefully that gets sorted because the value of like accounting for the biodiversity that we've got here in the Philippines is, I think, paramount, okay, and then of course, there's limited overlap in publication conferences venues between biologists, archaeologists, but I think what we are doing right now, okay, in this particular webinar is a step forward. Okay, now oceanic chickens, as I mentioned earlier, okay, we've been able to characterize them, okay, but in the Philippines, wala. But however, very recently, just before the pandemic, okay, so archaeologists coming from Cebu, okay, submitted archaeological chicken bones to me. Okay, so we are about to like process them. Okay, we haven't done so yet for various reasons. Okay, which I'm going to like share with everyone this noon. Okay, okay. So so one of the reasons why we haven't, okay, like for instance, processed the archaeological chicken bones is that one reason is that we really need an extremely clean lab to do that. Okay, now most of the samples that we process in the archaeological studies program are materials that are very low template DNA. Okay, so it means that they've got DNA in there, but they are vanishingly small. Okay, so it means that it is paramount for us to have a clean laboratory. So what we are working on right now are museum samples. Okay, but eventually, okay, if we are to proceed, okay, with ancient DNA work, okay, we need to have better laboratories for that. And then I'm sure everyone can imagine the capital cost needed to actually establish that laboratory. Okay, so in here, this is a spread of the work that we do in the archaeogenetics laboratory at the Archaeological Studies program. So the taxa that I'm most interested in, of course, includes the 4 commensals that I was mentioning earlier. Okay, and then right now with Ma'am Cora, okay, we're working on chickens, as you can see in this lower corner here. Okay, so the other taxa that I'm interested in are, of course, rats and, of course, the areca plant as well. Okay, so the potential, okay, of ancient DNA is unrealized, okay, in the Philippines at least, okay, but it has been demonstrated that we can actually get, okay, ancient DNA coming from tropical samples. Okay, there's been a lot of papers saying that, and that it's only really a matter of us, okay, like moving forward, okay, in actually doing that particular type of work. Okay, now in terms of like human genetics, okay, so it's very challenging because again, contamination reason. That's why we really need to have a proper laboratory, proper ancient DNA laboratory to be able to process any human materials. Okay, and then of course, until very recently, ancient eDNA, okay, ancient environmental or sedimentary DNA has proven its worth, okay, in terms of like accessing ancient DNA as well. So if anyone's interested, okay, I can pass you papers, okay, about ancient sedimentary or environmental DNA. Okay, so what we really need, okay, is a laboratory more or less looking like this, a very clean one, okay. Although ASP is taking steps, okay, towards that goal, okay. But however, you can imagine it's going to be very expensive to actually establish this, okay. And then of course, okay, capacity building, okay, for local researchers is of course very much in need, okay. But we are making small steps towards that, okay. But hopefully, okay, through this webinar, this noontime, okay, hopefully that with certain audiences, okay, that we can make that movement slightly faster, okay. All right. So now there are heaps and heaps of institutions that I'd like to say thank to. Okay, so of course the program on biodiversity, ethnicity, and forensics. Okay, IB as well. Okay, and then OVPAA, OVCRD, ASP. Okay, BR. So on and so forth. Okay, so thank you very much for listening this morning and for helping us with our work. Thank you very much. I think that's it.
Thank you very much, Dr. Herrera. Very, very interesting. When I first heard about the labuyo chicken, I really thought it's a spicy chicken. So anyway, it's a very interesting take on how migration happens, by not studying the people but the animals that they bring to with them. So parang nagsi-CSI talaga ano. So so before we wrap up, I would just like to call on our panelists. Before that, I would like to present a certificate of appreciation for Doc Mike Herrera. Maraming salamat sa for being a resource speaker during the webinar. And the previous speakers as well. Again, our webinar is entitled Focusing on Philippine Biodiversity, the start of the Philippine BioGenome Project. And I hope we host more sessions like this. And in the future, near future, hindi na webinar, makapag face-to-face na tayo. So we are going to open the next portion for our Q&A. We have received questions in our Q&A tab, and our panelists have been very systematic in answering them. Everyone can see the answers of our panelists for the questions that were posted. Also, we have seen some questions in our Facebook live stream, which I can share with the panel. So this is an opportunity for the panel to answer some questions fielded from Facebook. For the panel, first question. Is there any available local publication or articles we can look at supporting the decrease of baboy ramo due to African swine flu, ASF, or if none, may ongoing studies pa po?
Hello? I think sa akin ata naka-address yung question.
Yes po.
Okay, so thanks. Actually, yes, there is, meron ng existing na publication and na linabas yon last year. And yung publication is all about the eradication of the whole population. No, lahat ng baboy ramos sa isang forest ay naubos because of African swine fever. And ang reason nun, I think kasi yung kanilang forest or yung kanilang habitat ay nasa isang forest fragment. So wala silang matatakasan at napapalibutan na yon ng residential areas. Kung saan mayroong mga farming din ng mga domesticated animals. So it's so sad na I think that was the first record sa Mindanao, particularly in Mindanao Island, na eradication, 100% mortality due to ASF.
Okay, sir. Thank you so much for answering, sir. And a follow-up question pala, and I'm also curious about this. How do wild pigs get ASF when they are in wild areas? Paano po nagde-develop daw yung ASF?
Ang reason namin doon ay yung sa forest kasi yun nga may mga residential areas at minsan nakakapasok sila doon sa forest. And kung may dala silang pagkain, yun, na infected or contaminated ng virus ay pwedeng yun ang maging sanhi ng pagkalat. And ang baboy ramo kasi, kahit for example, magtae lang sila sa habitat, doon lang din sila matutulog or doon lang sila magstay. So mas mabilis yung pagkalat ng virus sa buong population. Lalong-lalo na, yun nga, restricted na yung kanilang populations. Maliit lang talaga siya na fragment sa Iho Forest.
And we have a question about whole genome sequencing in terms of marker identification. Have any of the researchers tried a different approach like ddRAD-seq instead of whole genome sequencing? Kasi mas mura daw po yun. ddRAD-seq. Anyone?
Actually, I'll just make a comment and maybe others could also help. Really, there are different technologies that could be used. So parang nga when we were inviting VP Bautista, ang kwentuhan namin, kasi syempre mahal, halos kalahating milyon lang yung bawat taxonomic unit. We don't envision in the program to sequence every single taxonomic unit. There will be representative. We have to set up a criteria kung alin ang pipiliin mo, kung ilan ang pipiliin mo, at kung saan manggagaling, ano yung representative. Parang ano, if you want to be very visual about it, sinong ilalagay natin sa Noah's Ark? Hindi naman lahat kakasya kay Noah's Ark. Sino lang ilalagay? So, this is why it has to be a staggered step by step. Kasi syempre, limited ang resources ng Pilipinas eh. So it could be that there are preliminary, and this could be a challenge also, could be something that your school or your university would like to be part of. Bago natin piliin kung sino yung whole genome, baka pwedeng i-DNA barcoding muna. Look at the extent of diversity using the simpler technologies or the faster, cheaper technologies. Once you have identified the degree of diversity, then that criterion, one based on your research, could already select the ones that will be used for whole genome sequencing. So hindi naman lahat. Thank you.
Okay. Thank you, Doc Cora. And while I was looking at the Q&A tab, and the panelists are also looking at it as well, what would you say are the biggest challenges we have in launching the Philippine BioGenome project para sa kaalam? If you can summarize it into two to three sentences in terms of infrastructure, in terms of human resources, what should be prioritized? Any from our panel?
Sige, ma'am, I'll try to answer that muna. Start it off.
Sige po.
Actually, the equipment, the infrastructure, we already have existing infrastructure, and there are already plans for expansion. I can only speak for PGC. I know that the Philippine Genome Center is already expanding. We already have facilities here in its main headquarters in Diliman. Although it's not the UP system, but it is housed in Diliman. And then we also have facilities and offices in UP Los Baños, UP Visayas, and UP Mindanao. And UP Los Baños, magkatayo na din sila ng building, hopefully. So, in terms of infrastructure, we're there already. And it's just a matter of continue applying for proposals to develop our infrastructure. What we need more are warm bodies. We need more people who will write the proposals. We need more people, experts, who will study these taxa and their genetic profiles, their whole genome sequences, and the omics that will give birth from all these genomic data. So we are also using this forum as an impetus for our future scientists, young scientists, to please consider taxonomy and molecular biology and bioinformatics as viable careers. So, yun lang po. Baka may gusto pa idagdag
May gusto pa po bang idagdag?
Dr. Christian Del Castillo.
Yes. As I mentioned earlier, in one of the challenges, I think the funding is a very big. We have the expertise, we have the facilities, but those still need some money to make them run, to be able to do the projects. Without that funding, all these ideas that we have, all these interesting things that we can do, we cannot do it.
Maraming salamat po, Dr. Del Castillo. And speaking of challenges, what are the positive outcomes you are starting to see in your research, considering po na we are parang hindi ko alam kung tail end of the pandemic pa? How was the research impacted by the last 2 years here in the Philippines? Were you able to see something na parang resource, parang resource na utilize nyo po ng maayos para i-share po natin sa mga gustong mag-research about flora and fauna? Paano nyo po nagawa yun na hindi nyo napupuntahan yung mga lugar na kailangan nyong i-research?
Are you asking me or everybody?
And everybody, yung mga experiences po nyo ng pagre-research sa pandemic.
I'd like to
I'd
like to answer
sige
that
po,
question.
sige po.
The last 2
Dr.
years,
Ginoo.
the last 2 years had been very challenging for us. So what we did, since we have a yearly monitoring program in the local municipalities here, so we just collected samples, and the challenge really is yung mga required tests na mag-negative ka. Because the municipality will not allow you to proceed pag positive yung. So there were quite a number of times that ma-postpone yung mga schedules because we have to have all our team members mag-negative sa test. But regarding the challenge also earlier, I'd like to concur with the idea of Dr. Carmelo and Dr. Ian. We really need the support of DOST and DNR as far as biodiversity studies are concerned. No, we cannot do things on our own. We need the funding of organizations like the ones that we mentioned. So I hope this will open doors through this webinar, that there will be more support from the government as well as from other organizations. That's from my side, Milet. Thank you.
Thank you, Dr. Ginoo. Dr. Bawanan?
Yes, may I also add, based on our experience, kasi we have this project, yung pabida ko, yung popularizing access to biodiversity information and conservation. Nung tamang-tama na matatapos na yung project namin, nagkaroon na nga ng pandemic. And so we were not able to really return to some of our research sites. So we really had to make use of the data that we were able to gather prior to the pandemic, and to be able to go back to the community to validate what we have, what our outputs, para to also secure their permission to share the output or disseminate the output. So kailangan naming mag-pass through the health protocols din. Kailangan negative ka nga sa swabbing. Pero luckily naman natapos pa rin namin, but hindi namin na-spend lahat nung fund kasi nga hindi na kami nakapag-travel sa ibang localities. And so we had to return it back to our funding agency. But on a positive note, may na-contribute din yung pandemic kasi isang example na rin to. When Dr. Ungria and Ian called us to meet and then start brainstorming about these things, about how we can collaborate. So parang it's a good exercise kasi somehow ay nawawiden yung ating opportunities, and we are also able to collaborate with other institutions and also have this opportunity to widen pa yung collaboration natin with the participants. And so that's on my end. Thank you.
Anything to add, Dr. Del Castillo, and after Dr. Monots?
Okay. From my end, what I noticed, I could point to two things. One is the normalization of genome and genomics. I remembered when we were making a proposal pre-pandemic on genome, and the reaction of the GC was, genomics, ano ba yan? Ano, mapera ba yan? Ayan. That was the reaction. So after the COVID, I passed the same proposal, and this time they were more amenable to a genomics approach. Because of the COVID, I think the genomics idea became more mainstream and more acceptable to funding agencies. Another one is this Skype or Zoom rather. It shows ang age ko, no? Mga Skype yan ako. So it shows a bit. So Zoom makes collaborations like this a bit easier. Of course, meeting each other in person and then and talking with each other is much better. But this helps a bit in in making us engage with each other in a more faster way. It's not easy for me to go to Manila if I don't have a project, because I would have to foot the bill myself. So having to meet, for example, personally, going to Manila to meet personally, let's say once every month, like that, if I don't have a project, that's not an easy thing to do. But with Zoom, hey, let's meet again next week. Let's meet every day if you want. Okay, it makes those things easier in this regard. And may I also just add that there's a question from Ms. Vivian Panes. Maybe you can add that later if after the reply of Dr. Monatilla. I think this is an important question from Ma'am Vivian.
Thank you, sir. Dr. Monatilla, yes?
Actually, in relation sa sa comment ni Dr. Castillo, medyo challenge din yung regular na Zoom meeting at tapos ang nakakawindang yung preparation ng proposal. Because you have to make your storyline, you have to collaborate a lot of people to be a member of your team. Tapos at the end of the day, disapprove naman ng DOST. Kaya parang nakaka-disappoint. I don't know how we can resolve that. Kasi yung area naman natin, yung mga evaluator, asking the evaluator, talking to the evaluator. Tulad niyan, ang sa aming program, 3 projects. Bakit iisang project lang ang kinausap? Hindi ang whole, ang ibang projects, ang 2 projects ng program. Tapos in limbo kami on what we will do kasi wala namang information sa amin. Walang feedback.
Dr. Monots, I will set a meeting for the program and we will discuss the repercussion of that development. Meron pa tayong sagot. Sir, we are scientists and we are creative. We will create solutions, sir.
Yeah.
Yes. Wag kang bibigay, sir.
Thank you, ma'am.
Welcome po.
For the panel and suggested by Dr.
I'm sorry. Sir Monots, just to repackage and then change the title a bit. Pwede na yan.
Ayun. So for the audience, no, nag-share na yung mga researchers ng mga challenges nila from creating the story in their research proposals and all the blockages that they have. So I have read a very specific question here that has been subject to a lot of researchers. Hindi naman worst nightmare, pero ready sila palagi sa ganito. Parang sabi dito, as I listened to our previous speakers, 2 of them submitted proposals to DOST in whole genome projects kasi yun yung call for proposals. However, their proposals were disproved, which is lahat parang pinaghirapan, nadidisappoint. Thus, I would like to know if DOST has a priority list of species for the whole genome project. It will be good to know their priority list of species so that we can refer to this list when we decide to write the proposal. Parang, I'm pretty sure by the end, before the year ends, magkakaroon po ng maraming research write-shop po ang different institutes kasi yan po ang first step sa pagkakaroon ng maraming research approvals. So who would like to share parang ano yung insight po nila dito kung may list po ba? May database po ba? Ano po bang priority natin?
Actually, I'll butt in. Ian, tulungan mo na lang ako. All right. So, and I think it might be an idea now to talk a little bit about what the program on biodiversity, ethnicity, and forensics, what are the plans? Kasi syempre, we said earlier, and I actually, that's one of the reasons why we have actually asked Dr. Iborra to provide the opening remarks, and also VP Cynthia Bautista, because as we know, and that's what's been said here, some of our policymakers or our funding agencies may be a bit more distant to the ground being us picking up whatever organism you would like to study. So this is to actually have that exchange of information. So ito po yung plano namin. And again, I'm not promising, but I would really invite people to be part of the planning and the implementation. So if you have ideas, we've given you a contact point. In Raiki is a Master of Science graduate. He's with the CIP program of the Science Education Institute, and he's working full-time 100% on the program on biodiversity, ethnicity, and forensics. So we're trying to find creative solutions to many of these challenges. I wouldn't call them problems, but challenges. So ang unang punto, kaya tayo hindi pa nakapasok kay Earth BioGenome Project. And this is, gagawin namin tong springboard to get funding. Hindi tayo makapasok sa global kasi ang Philippines has not provided us enough support to be able to be part of the global initiative. So we need funding for at least some of the projects. That is it sounds bad, pero gagamitin natin siya para yan ang, you want us to do global work, you give us money to do global work. First point. The second point is what we're really pushing with the program on biodiversity, ethnicity, and forensics. And for others, you might have heard me talk about this on the forensics part. Now, medyo po kasi ang nangyari sa programa ng PGC para at least maintindihan lang na iba. Before, biodiversity and forensics were different. Yung sa humans, ethnicity, forensics kanya. Yung biodiversity kanya. Pero ang ginawa ng PGC, puro kayo biodiversity, kahit anong hayop, kahit anong tao, kahit anong halaman, biodiversity pa rin. Ipagsama natin. So which is why we now have a program on biodiversity, ethnicity, and forensics. So with a team of the Natural Sciences Research Institute, headed by two of our senior scientists, Frederick Delfin and Jaslyn Salvador, has already been networking with a lot of state universities and colleges on the work with humans, on the indigenous people work, etc., etc. You might have encountered them. Now, here we're trying to bring in a similar concepto, which actually PGC Visayas and PGC Mindanao have already their Mindanao Network and Visayas Network Consortium. What we really need, and I've heard DOST talk about this, is we need to show that we are very productive. We need to be more open to collaborate, cooperate, and communicate. Pasensya na, Cora, kasi. So, collaborate, cooperate, and communicate, lahat para lang daw ma-maximize yung pondo. This was what we experienced in forensics before. When we started with the human staff, talagang walang nagpopondo sa amin. Now we have been given 2 large funding for 2 programs already. And in the last defense, we were being pushed to go, to submit, to modify, to amend, but it looked very positive. So this is what we want to implement with the biodiversity program, similar. Collaborate, communicate, and cooperate. So it shall be a consortium. So this is why we are discussing this, and we need to support each other. We need to create a track record. Kaya tayo hindi napopondohan kasi pa isa-isa tayong nakikipag-usap. What we think, and this is the work that Ian and the team will be doing here, sabay-sabay nating kausapin. Sabay-sabay, iba-ibang angulo. And this is why this webinar is fantastic, because before ay puro pagkain lang pinag-uusapan ninyo, ay hindi, puro ganyan lang pinag-uusapan nyo, ay dahil meron kang maliit na farm dyan, gusto mo yan ng aralin mo. No. It's really for a national perspective in terms of conservation, in terms of agriculture, in terms of our human and animal history, in terms of resources that we can use to make things more useful for humans and animals and the earth, in the environment. So yun po, yung nakikita ko doon, if you email in Raiki, give your contact details. I'm conceptualizing a possibility of a mailing list or something like a resource network. Para doon kayo pupunta, pag meron kayo i-check, silipin nyo na lang doon or put it in your favorite Facebook, whatever. Anyway, we'll talk with PGC, etc. But communication is an integral part of this. And then po, pwede nating, nakinig ako kay VP, hiningi ko actually yung talk na kanina. We really need to start the process. Because for funding to be given to us, we need to establish a track record and we need to show the scope. So kayo po, I think the participants here, you will be the partners in terms of making sure that the scope is not only UP Diliman. It's not only this little dot on the map. Ang laki-laki ng mapa, not this little dot. So, for example, you can, the state universities and colleges or any university, private universities for that matter, could already identify the taxonomic unit that you want. Naghanap kayo ng criteria. Actually, we tell them what we need. How do you tell them? And this is something that we did during the last 2 years. Some of the team that I've had, nag-review of related literature, walang magawa sa labas eh. Sabi ko, kaysa mag-isip-isip ka ng kung ano-ano, review of related literature will make it compelling and convincing that this particular animal or plant is worthwhile studying. And the only way we can do this is we give the facts. We are updated in our information. Para pong, pasensya na, paminsan nasa korte ako, we need to be so compelling that the judge will believe us kahit nanginginig na yung tuhod mo sa ilalim ng mesa. Nanginginig na yan, pero you're so compelling and so convincing. So in fact, at this point, I'd like to invite you, kasi kami nagko-conceptualize, mini-message ko si Dr. Ian eh. We will come up, si Dr. Ian ang editor ng Diliman Review. So kinumbinsi ko na siya that we will have a special issue on biodiversity. And of course, we're inviting the speakers here. There were many questions. Pwede ba naming makuha, mahingi yung slides? Papasulat natin yan sa mga speakers. It's your review. What you've talked about, can you write a review on it? And then I've got an illustrator. If you want some of your illustrations to be re-illustrated, para walang copyright issues, papa-illustrate natin yan. Gagawin natin yan. Pagkatapos, and others who are in this webinar, maski hindi kayo speakers, but you think you've got your favorite animal around there in your probinsya or your city, write a review. Submit it to the email in Reiki. Pero Diliman Review yan, di ba, Ian?
Yes, ma'am.
Yung website. Yung website. Ang deadline ko, nilagay ko June 30. Send your review. We'll get it. We'll help you in terms of the editing, etc., etc. If needed, if not, we just send it to the review. And then it's your contribution to getting your taxonomic unit in. Bakit? Pag na-publish ang Diliman Review, Ian, yan ang ipamimigay natin sa DOST. Pag nailagay niyo yung hayop niyo dyan, nasa listahan niya ng DOST. So, ibig sabihin, meron na kayong one foot in. Pangalawa is also, we are already conceptualizing other possible funding sources. I've looked at the Australian Embassy's webpage. Actually, the Australian Embassy and other foundations, I think some of you, I think Leaf also showed some of the foundations that have their pet in mind or animal. So if not the OSC, will have to look. So we need to be creative there in that sense. Dr. Cynthia talked about curriculum development. And that's something we need to conceptualize. We've got a team of people here. Actually, we were thinking about it before, pero ngayon meron na tayong Zoom platform. Ian, before po kasi kami ni Ian Fontanellia, we had the forensic seminar. Sa Diliman lang, hindi makapunta yung iba. So we kept on being invited. Sabi ko, Ian, i-modular na nga natin to. Such that, bibit-bitin natin lahat. Pupunta tayo kung saan bit-bit na natin lahat yung speakers. But now that we have the Zoom platform, which is, I guess you look at the positive side of something that was negative, then it has given us a platform. So ang ating communication, I agree, physical reporting, lakwatsyera po kasi ako, so mahilig kaming magpupunta kung saan-saan. Pero at least with a blended communication, with a Zoom or Microsoft Teams platform or whatever platform we've got, then we are able to reach a lot more people. So we've done this, I think with NCIP, ang ginawa namin with Facebook, I don't know if you have opportunities. Actually, nagkakaroon ng data even with Facebook, pinapa-data lang, etc., etc. in some of the key sites with proper notification. So we might be able to do this in terms of curriculum development. Hindi naman kami makapag-bioinformatics, Dr. Ian or Dr. Ganyan, kasi hindi na pa namin maintindihan. O di sige, we go a few steps before. So that is that we will be able to do that. And we have partners here. We have people here in Visayas. Ian doesn't need to go to Visayas. Andiyan na si Dr. Del Castillo. Ayan na, oh. Kidnapin niyo si Professor Leaf. Andiyan na sa Mindanao. Dalhin niyo na ako sa... there are people there. It doesn't have to come from Diliman. We are now. And we've got PGC Visayas and we've got PGC Mindanao. And then lastly, ito I open na the invitation. Our next webinar is on policies and research integrity. It will be on May 27, again, run by this program. One of the things that came out in the I think Ian had it in his slides, is step-by-step yan eh. So that in a step-by-step, one of the things na medyo challenging is trying to get those permits. The permits from the ENR, permits from ethics. Wala kaming ethics dito sa probinsya ng malayong patunguhan, wherever probinsya. Wala kaming ethics dyan. And I've already been asking ethics here in Manila. Baka pwede naman i-adopt nyo yung ibang region. Kung halimbawa sa Region 2 walang ethics review ko, baka naman si Region 2 pwede mag-submit sa Region 3. Baka naman pwede mag-submit sa Region 4 kasi wala pa silang ethics research com. So those problems, sasabihin, wala kaming magagawa. Hindi, meron tayong magagawa if we work together. So these are things that we would like to discuss on May 27. I'm writing the concept paper now. We're writing the concept paper now within Reiki. And then we will invite everybody on policies. And one thing that is clear na lumabas dito sa discussion natin, promise huli na to, is that sinabi ata ni Dr. Del Castillo to inuunahan tayo ng mga taga-ibang bansa. Pupunta sila dito, hello. Pag uwi nila, meron na silang isang crate ng samples na whole genome crate, exagerada talaga. Isang crate ng samples na iuwi na nila without a material transfer agreement. Now, we've encountered that. You may have heard me in other platforms. We've encountered that in several occasions already. So what we want to do in this program is to make, in the research integrity, is to show or to find out how often does this happen. Kasi kung ako lang or one person lang, sasabihin, Cora, sa'yo lang nangyari yan, di naman yan. But if we can show the institutions that this is more widespread than what we know it to be, then there is compulsion, we will make it compelling, such as what Indonesia did, for the institutions to address the problem. Whether it's through policy formulation, whether it's be legislation, whether increased surveillance of the activities of other researchers in the Philippines, whether foreign or local. And it's just also to help our local researchers kasi matatalo talaga tayo as Dr. Del Castillo nga sabi, matatalo talaga tayo, bilis nilang tumakbo eh. But we are not helping the Philippines improve in terms of human and human resources and infrastructure. So ito po yung mga plano natin. And then also para lang this national consortium, ito ang gagawin natin plataforma, iyan. Narinig ko na DOST. Gusto nila ng national consortium. In fact, there are plans for biobanks and PGC will be an integral partner in biobanks and data resources. May nagtanong kanina sa question and answer, Ma'am, pag nilagay mo lahat yung whole genome sequence, di kukunin na ng ibang tao, yan gagawa na silang kung ano-anong gamot, talo na naman ng Pilipinas. Wait lang. Hindi ko naman sinabing pag nilagay sa data resource, everybody has access. Lalagyan ko ng filter. Hindi pala ako si Dr. John Yap. Lalagyan ng filter yan. So really there are, and that's what we need to formulate policies. Who has data access? Because that is based, particularly for humans, based on the informed consent. Based on the informed consent of the people who owns the DNA. We do not own the DNA of any individual. It is the human source. We are only custodians. And that is a big responsibility because we need to be responsible custodians. That's when ethics will come in. So, ang plano po namin, medyo mahaba-habang plano na is how do we make it simpler in the program, how we make it simpler. Kung isang template lang naman niya, Ma'am, ethics review, paano ba sinusulat yan? Tutulungan namin kayo. Meron na kami mga templates. Palipalitan nyo na lang yung pangalan ng hayop o ano yan. You know, those are things that we can help each other so that you don't start from zero. Pag kayo naman may nagawa at maganda, i-share din naman natin sa iba. I think that is something that we would like to encourage in the program. And so kahit medyo, Dr. Monots, medyo na-disappoint ka, hindi naman disprove, disapprove. Disprove is different. There's factual information there. Disapprove. Pero liligawan pa rin namin sila. Magaling naman si Dr. Ian manligaw. O di siya, di yan ang gagawin natin with the
Thank you, ma'am.
Alright.
Ang hirap ng 1 month na
yes,
once sa 1 month, i-build mo yung team mo tapos gagawin ng proposal.
Sir, ang ginagawa lang,
magigising ka para mag-upload.
Hindi po yun lang. So, binabali-baliktad lang yan. Parang ano lang, binibihis-bihisan lang yan kung ano-ano. Pero we actually improve. We will improve it, sir. We will improve it. Nakatasan kami ni Dr. Del Castillo. I'll be quiet now. Thank you very much to everyone who attended and our speakers.
Dr. Del Castillo, you have something to add?
Yes, 2 points, no? One, ang continuations, again mentioned ni Dr. Ongria about owning. It's not necessarily owning the genome, no? But this has not happened yet. But personally, I foresee in some future scenario wherein someone who sequenced the genome first will have first dibs at whatever uses of a genome there. Okay. Example, some other country would be able to sequence the genome from a plant in the Philippines that has a medicinal property. Okay. So this has not come up yet, but it's not unforeseeable that in the future that this can become some sort of intellectual property of theirs. So if we do not have this, we're not able to sequence this first, we will be left behind. Okay, so this is, again, as I said, this has not happened yet, but these are not impossible in the future to happen. Okay? And then another point, back to that question that if there are priority species, as far as I know, in that there is no whole genome project yet of DOST, so there's no priority list of species for whole genome project. But DOST, as far as I know, has priority species, okay, mostly of those of commercial value. As in the aquaculture field, we usually pass projects in milkfish, in tilapia, in shrimp. So DOST usually has those priority that if you're not there, they're less likely to even look at your project. They look at it and then just maybe, So I'm not in the selection process, so maybe this has to be addressed. That project, what is the government agencies, DOST, DNR, DA, maybe they have to consider that. Well, whole genome sequencing projects would most likely not be in one of the priority species, because most of the time those priority species already have a whole genome sequence. Okay, so I think, ma'am, we have to put that into consideration.
Yes, sir. Dr. Del Castillo, one creative way we found there is nagpapa-adapt kami. So kung meron kang 5 hayop, halimbawa, you are interested with something, hanap ka ng 3 dyan na nasa priority. Yung 2 nasa bandang likod. I mean, i-declare mo. Pero nasa bandang likod sila.
Yeah.
Diba?
That's an interesting
parang
approach.
Lahat, yung 96 well plate mo, may nakasabit siyang 48 na wala sa priority. Na nagagawa mo rin. Pero it just has to be clever in the way things work out. And of course, those solutions we can share. No problem po.
Thank you for that hint.
Anyway, for our panelists, Doc Ian, sige po.
May sinabi lang meron sa mga participants natin from NISMED, UPDB, National Institute for Science and Math Education and Development. They have sana many daw educational components in project. not just for the tertiary or postgraduate level, sana sa primary, secondary, SHS, ma-disseminate na rin ito in order to, communicate effectively. Ito yung part ng communicate na sinasabi Dr. De Ungria. We start them young so that we will have warm bodies that will eventually succeed us. So para ma-generate din yung interest at yung ma-inculcate din yung importance ng ginagawa natin. So thank you very much to Ms. Med for suggesting that and for helping us as well. Sige po. Yun lang po.
Okay. Maraming salamat, Doc Ian. I think that's all the time we have for the Q&A. And while the panelists are there, we would like for them to open the camera and we will take this opportunity to get our group picture, yung ating documentation na nag-webinar tayo with the panelists.
Alright po, I'll be taking the photo. And 1, 2, 3.
Smile.
Okay. Isa pa po for safety. Alright, 1, 2, 3. Alright, thank you po.
For those who have posted questions in the Q&A, after this, the panelists will still, if they have time, hindi na po natin i-discuss sa Q&A, baka po mabalikan pa nila yung chat tab, hindi pa po namin isasara yon or yung Q&A tab para makita po nila yung mga additional questions na nagsulpot. And please also check the chat box for the link regarding the feedback for this session. And to close our webinar, I would like to call again our superhero palagi na lang si Doc Ian, ang tagasara ng pintuan. You have the floor again, sir, to wrap things up.
Okay. Thank you, Shelette. Actually, I'm going to read the closing remarks in behalf of our Executive Director of the Philippine Genome Center, Dr. Cynthia Saloma. So, good afternoon, everyone, and thank you for joining us today. And I would like to believe that today's gathering signifies our common interest to protect and conserve the Philippines' rich biodiversity. Now, last June, Dr. Saloma received an email invitation from the executive manager of Earth BioGenome Project in UC Davis if the Philippine Genome Center is interested in becoming an affiliate project of the Earth BioGenome Project. Hence, the generated interest and the impetus, one of the main impetus for this networking. So, take note that the EBP is also aligned with the objectives and principles of the Nagoya Protocol on access to genetic resources and the fiat and equitable sharing of benefits arising from the utilization of genomic information. Now, as a molecular biologist, researcher, and as Executive Director of the Philippine Genome Center, Dr. Saloma cannot help but get excited to receive such an invitation. So the participation of the Philippines in this endeavor will definitely highlight the richness of the country's biodiversity in keeping with the goals of the Earth BioGenome Project and help understand the evolutionary relationships of all organisms, discover new species, elucidate genome evolution, and develop evidence-based conservation strategies for endangered and rare species in the country. Now, this is our chance to contribute to the global effort to sequence all eukaryotic life, multicellular eukaryotic life on the planet in the next 10 years. So we just hope that the Philippines will be a major contributor to this endeavor. So allow Dr. Saloma would like to thank the Biodiversity, Ethnicity, and Forensic Program Director, Dr. Cora de Ungria, for leading the team to put together the Philippine BioGenome Project, or PBGP, which aims to establish a national collaborative network of universities and colleges and government agencies to sequence and catalog the complete genome of selected Philippine taxa. So we also note amongst the audience the participation of even NGOs like the Katala Foundation, the yung sa Maubaya Foundation, all the other NGOs, the Philippine Eagle Center. So we are also going to tap these as our partners in this endeavor. So hopefully, and also the Energy Development Corporation. And also, I think we have representatives from the Department of Science and Technology and even the Department of Agriculture, as well as the Department of Environment and Natural Resources. So we are all going to be partners here. Hopefully, to this webinar, more institutions outside UP will join us in this endeavor. Again, in behalf of Dr. Saloma, we thank our guest speakers, audience, staff, Dr. Diungri, our Program Director for Biodiversity, Ethnicity, and Forensics, for successfully holding this webinar. So maraming salamat po sa lahat, and I hope you enjoy your late lunch. Mag-uusap-usap ulit tayo. We shall continue this conversation. Yun po.
Thank you very much and have a great day. Thank you.
Thank you so much to our panelists. And we would like to remind everybody to check the link for the feedback form in chat. It's also posted on our screen po. We will be basing the names of your attendants on the answers of that feedback form. So do make sure that you have spelled your names correctly. That would be for our reference. And of course, magpo-promote po kami ng upcoming, ni naman po movies, no? Pero nagki-kick off na po kami ng maraming webinars, possible write-shops for the program for biodiversity. So, sana kita-kits ulit tayo sa May 27. Nabanggit na po ni Doc Cora to, inuulit ko lang po. Research governance in the Philippines. If you are, sabi nga po nila, one of the things that we need to really work on is to consolidate all our resources para magkaroon tayo ng global voice pagdating sa research at makasali na tayo sa Global Biogenome Project. Research governance in the Philippines, May 27. More information will be provided in our Facebook page and other social media sites. Don't forget to like our Facebook page. So, magandang hapon po ulit sa lahat. Sana po masarap ang lunch natin. And that concludes our webinar entitled Focusing on Philippine Biodiversity, the Philippine BioGenome Project, brought to you by the Program for Biodiversity, Ethnicity, and Forensics by the Philippine Genome Center and UP Diliman's Institute of Biology. Thank you, maraming salamat po at kita-kita ulit.
Maraming salamat sa lahat.
Bye-bye.
Bye-bye. Thank you.
Thank you.
Thank you, PGC admin, Shilette, Rachel, and Raiki, Ariel. Thank you.
Salamat po, ma'am. Ingat po.
Kain na kayo.
Salamat po
for the planning.